Okay, this is complete IMAT Cell Biology, in which I have talked about all the organelles. Actually, this video I have done a live class for IMAT Accelerator students. So this video is going to be very comprehensive. In first 10 minutes of video, you are going to see which chapters are very important in all cell IMAT biology. I have talked about from previous year, this number of questions have come and all that stuff. And then later on there is complete overview about IMAT Cell Biology. We have talked about all organelles, and in last 10 to 20 minutes we have discussed the past paper questions from this cell biology. They have asked these questions, so we have done like 5 to 7 questions. So that video is going to be very helpful for you if you are planning for IMAT, because as you know, IMAT cell biology, actually cell biology is just constitute about like 11 to 13%. 13% means that there are at least 2 to 3 questions every year from cell biology. So that's why you have to do that, and it's easy, okay, it's not hard. But before moving on to video, I want you to attempt past paper quiz. Okay, those questions that we are going to attempt in the end of the video. I have got a Google form in below video description. You must attempt that so that you can realize how much you are scoring. Those are literally all the past paper questions. Okay, I have just copy pasted from IMAT past papers. Just the purpose is to give you a realization to idea about what you are going to learn, like what is your level before the lecture and what would be your level after the lecture when we will be discussing that. That is very comprehensive video. We have talked about everything from cell biology, all the organelles, all the differences, and all the past things that IMAT has asked. So that is video is going to be very helpful for you, although it is very lengthy. So I would say watch it, that will be helpful for you. And I have got all the lectures just like this for all subjects like chemistry, biology, physics, that are very comprehensive, like 2 to 3 hours for each chapter. But just watch it and give me feedback in video description. So I hope you will enjoy it. So start. First 10 minutes is comprehensive overview of IMAT biology, which chapters are very important. You have to do all that thing, and then in next we have got all the cell biology, and then at the end there is past paper quiz that we have discussed. Okay, that is the same quiz that I have given in the video description, so must attempt that so that you can like enjoy. So just begin.
So today we have got our first day. So we are going to do our first lecture of on academic lecture that is supposed to be. Yesterday was supposed to be orientation, and in that orientation we did discuss a lot of very cool things, and I hope you enjoyed that. So today we are going to firstly we have got on the plate cell biology. Okay, we will discuss that. But before moving on to that, you must know about biology specifications for IMAT. So I want to give you a complete overview about biology specifications for IMAT. So you must like know them and what are they, how you are going to study them. There are like few chapters that I have made and I have categorized them. So I think they are going to be like super good for you. So let's start. First, we are going to knock down the whole syllabus. Not syllabus. I want to get you what is IMAT syllabus for biology, because today we are starting biology. It's cell biology. So I want you to know that what is in biology. So you like try to hear what's in biology. Okay. So just see it, what's in biology. So just let me share my screen and then we will cover up what's in biology. I have made this whole masterclass on YouTube as well. So I hope you guys like have viewed it, and it's you know about these things. But anyways, I want you to like introduce you with like these are the things that has been mentioned in the IMAT 2023 syllabus, because there is still no IMAT 2024 syllabus yet out, and I am now believing it will be out. So just let's talk about. You are going to first of all, okay, this is the easiest thing you can make yourself for yourself. Like I have made very easy table of contents. You can utilize them. You can make your own planner sort of thing. First of all, the first chapter that we are going to see is cell biology. Okay, cell biology, it's like it's not hard. It has got organelles, but the cool thing about this, it has been asked in every year. Okay, every year of IMAT there was question from cell biology, so that's very important. And second one is going to be biochemistry, and biochemistry is very important. Every year there are 2 to 3 questions. Okay, 2 to 3 questions. So I think that's like that makes sense. Not two, but at least two, one, two. Maybe write it one, two. Okay? Then it will be like more clear. 1, 2 question every year. And in bioenergetics is very good chapter. Although most students fear like it has a Krebs cycle, electron transport chain, and other thing. They think that this is like sort of a hard chapter. No, it's very easy chapter. So, but bioenergetics is important because Here cellular respiration is very important. Glycolysis, all the like steps, glycolysis, then Krebs cycle, then there will be electron transport chain. In 2023 there was question from bioenergetics in biology, one question. So now you can understand how important it is. Okay, so it is so important. So bioenergetic is sort of important. So bioenergetics is super like good one, but here you have to memorize tons of information. So this is also included in this. We will study photosynthesis, and second thing we will do respiration, cellular respiration. That is like a very important thing. Then cell cycle. Cell cycle is very good one, and it has got mitosis and meiosis. But there are so many tricks in it. So you guys have to understand about this. Then genetics. Genetics is the major portion of IMAT biology. There are at least 3 to 4 questions every year. Sorry, 3 to 4 questions. Believe me, 3 to 4 questions. There are just two like sort of genetics. I don't know. I have added inheritance as well. No, it's included in inheritance. So it will be one is going to be inheritance. Okay, inheritance, and second would be molecular genetics, and these are both very important. And mutations is going to be like important, because 2023 and 2023 I'm at there was one question from mutations, so I hope you guys can understand it. Then there is evolution, and in evolution in 2023 there was one more question from Hardy-Weinberg theorem. Okay, Hardy-Weinberg theorem. Okay, biotechnology not that important, but we will be doing that. Okay, not that important. But anatomy and physiology, it's very like important one, and in that I have categorized in whole week schedule for you, but let me write here for you what is included in it. Okay, so first of all, as you guys know, all the like all the systems, for example, digestive system. First of all, that is very important. Digestive. Then there is respiratory system. Respiratory system is super important. Then there is going to be immune system. Okay, immune system. After immune system, what are there? Reproductive system. Reproductive system. Okay, reproductive system. After reproductive system, there is one more that is supposed to be, what is it? Is there anything left? There is a lymphatic system. After lymphatic system, there is cardiac system. Cardiac system. Is there any left? Circulatory. Yeah, circulatory has got lymphatic and cardiac system. Cardiac. So I just call it as circulatory is neuro system, nervous system. Yeah, I haven't like included this. Oh, that's very bad. Although it's going to be, yeah, that's going to be in this nervous plus hormones. I call them hormones, but you can urinary system is included in homeostasis. Okay, urinary system. Urinary is in here, nervous system is here, and the second one is going to be your hormone that we call as chemical coordination. Okay, nervous coordination and chemical coordination, but I would like to write them as hormones. Okay, as good. But these are all okay. These are very important, so we have to cover them. It means that biology has got 23 questions in IMAT. This is the like a big syllabus that biology covers. I hope you guys got it. And animal tissues, there is going to be epithelium. Okay, epithelium. As I know, you guys just I'm like want to say to you, just get into med school. There will be like 3 months. There will be 3 months. Then that you will study just epithelium. And then you said that that's very good. I have added skeletal system. Skeletal is also added. Skeleton and muscles. Skeleton plus muscles are also there. Yeah, sorry. Thank you. I know for like letting me know. This is just IMAT syllabus. This is just a little syllabus. Okay, this is just little syllabus. I hope you guys got this. And right now, let's move back to our today's session. That is going to be cell biology, and we will try to cover it up ASAP, because I want it to be like very. We can cover it early, then we can talk. Okay, that is like going to be some little bit good talk. So now understand this thing, and you guys can ask any question during class. Okay, don't worry. So first of all, let's talk about cell. And before moving on to cell, remember this: those students who draw spider diagrams, mnemonics, they are going to perform well in the IMAT. Otherwise, that will be very hard for you to remember all of the information in this short period of time. Remember this, my phrase. Okay, those who done smart work, okay, those who done smart work, they are going to perform well in IMAT. Those who just like put hard work, maybe just passive things, pick a book, read it, pick up notes, read it, and then attempt mock exam. That's all. If you did that, you are not going to like perform very very like great that you have expected in your mind. Because I tell you guys, I told you yesterday, time is very less. You will have to increase your study hours, and competition is very very high. Okay, very very high. I hope you guys know these things. So let's talk about first of all what we are going to do study today. That is first of all the big thing that we have got. Okay, that is we will cover. Okay, first of all the cell theory. First of all, we will talk about what is cell. Okay, what is cell? This is going to be today. Firstly, today's chapter, and then there is going to be the second important thing. That is going to be cell theory, because in IMAT past paper, okay, they have asked questions from past cell theory. Just you can believe me, it has been asked in past papers. Cell theory. That is like Schiller and Schwann given just 3 to 4 points, and they asked from they made question from there. So like you can see how is IMAT. It's more very very specific, and it's very very mad. Okay. So see here, after cell theory, we will just straightforwardly go to organelles. Okay, organelles. But before organelles, the major and important structures are cell wall, cell membrane, and cell wall. Out of them, cell wall. Okay, cell wall, cell membrane. Cell membrane is super super important. Every second year or every third year, they make question from cell membrane. I have given you question in the past paper quiz that I hope you guys solved it. I let me check. Okay, 14 students have solved that, and out of like I hope you guys got the idea. After cell membrane, there is going to be organelles, and organelles are super super important. Super super important. I have like written in the morning. I told you in this mitochondria, then there is going to be so then there is going to be the after mitochondria there is going to be the Golgi, then there is mitochondria, Golgi body, then there will be endoplasmic reticulum. The nucleus is super important. Okay, nucleus is very very important. Then there will be plastids, and in plastid chloroplast is very very important. After plastids there will be different organelles. Ribosome is very very important. Ribosome, then there will be lysosome. Then there will be what are is there any left? Yeah, we can cover them. And then there will be in organelles. After organelles, there will be a very important topic that I want to let you know is that animal versus plant cell. That is very very important debate, and they have clearly mentioned in the IMAT syllabus. Okay, animal versus plant cells. Animals versus plant cell. There is just supposed to be difference, and then there will be prokaryotes versus eukaryotes. Prokaryotes versus eukaryotes. That is like very very good things, and you can just read them. Okay, cytoskeleton. Yeah, cytoskeleton is also included. You will be reading it from your books. Okay, I don't have got in the notes. You will be reading it from your books. That is cytoskeleton is also included. You will have to know about you have to know about what tubules are tubules, and what is the composition of tubules. Tubulin protein. Then there will be other spindle fibers originate from origination. Also, we will talk about them. Chloroplast, yeah, mitochondria is here, iron, and plastids has got chloroplast. Okay, chloroplast, chromo, and the third one is leucoplast. Okay, so we will tell you about this. Now I hope you guys got the overview. Although I have sent it, we will not be doing every day. Okay, because I just Vacuole is also there. Vacuole is also there. Yeah. I have written everything, but now I can't like recall it. Like, the fuel is also there. The fuel is very important, and in nucleus we will also be studying this. These what it is the important one? Chromosome. Chromosome is very important. Okay, so we will also be doing that. I hope you guys are getting these things. Yeah, there are like tons of other things that we will cover them up. Okay, so just this is the thing that we have to say. So, and I have sent all these things in the morning. I will not be sending. I will not be doing every day because we don't have time. This is the first day, and most of students couldn't didn't know where I place, how it's been done. So let me share my screen and let me know how I did that. Okay, so you guys can like see it, and I will not be doing every day for this. So just see this screen. Can you see my this Discord screen? Just wait a second. Can you see it? Can you see my Discord screen? Okay. Now see this here. Every day there will be daily schedule. Okay, daily schedule. And in daily schedule, I will be sending good morning. So you can like say in your heart like good morning. Okay. So here then there will be whatever you have to cover in today's cell biology chapter will be mentioned, everything will be mentioned what you have to study. Okay, and I do write about these are the super important things, and I do write some short summary notes. I have written these from like ages here, so you can study about. And what you were talking about cytoskeleton, so I told you micropyramid, intermediate pyramids are very important. So you will study about it. And then there will be passive transport is also important, and active transport, endocytosis and exocytosis, we will be covering them. So I will give you overview, but for good reading, you will be reading any book. If you guys don't have books, okay, PDFs of book, I will share them in here, okay, resource section. I have shared my notes here. You can just simply click or go and download them, and I will share all the books here. So those who don't have books in the short form, they can use these books. I hope you are understanding what I'm trying to say. So let me share my screen again. So I hope it's going to be like a good thing. So let's talk about cell biology. Let's go to that. We have to do all these things today, and that's like very hard. Okay, so let's do it. Okay, do it. So see here. First of all, let's talk about cell, and I will talk about this after this lesson. I know this is going to be very basic, and I will talk to you in the Discord that what is going to be the style of lecture, because I don't want to focus on basics, because basics are, if you focus on basics, that is because you guys all have got knowledge from your high school, and you know pretty much more than me about cell, organelles, all those things. My idea is to discuss questions, how questions can be made from here. So more discuss mock exams, and then give you an overview what you should be reading in this specific chapter. Okay, so that's like a good thing for you that will help us to be more productive and do more. But don't worry, we will discussing everything in lecture. Okay, it's not a thing I will like exclude it. No, we will be doing, but we will be like focusing more on prominent things, more on more productive things that you guys can do. Okay, which I can like give you guidance that I should be doing that. No worry, we will discuss about those things. So first of all, let's talk about cell. What is cell? Cell is just a fundamental unit of life. Just if you guys know that we have got the walls that are made up of bricks. Okay, bricks. So that is their fundamental unit. And for humans, the fundamental unit from which we are made, it is cell. Okay, cell is structure and functional unit. Okay, structural and function. It has got some specific structure and it is functional. Okay, it has got some very great function. So let me just make it a white. I think make white more great. So microscope, this is just a discovery. They don't ask this type of discovery. You can study it from here. I have written in like very detail. So you guys can study it. Just wait a moment. I can like make it. You guys can study it. It's like these discoveries are not that important, but what is important that yeah, these are not important. Let's talk about first of all cell theory. What is cell theory? So Sheridan and Schwann, there were two scientists who have given this theory. Okay, Sheridan was zoologist, and Schwann was botanist. So I think Schwann was zoologist. I don't know, whatever it is, you can like do this thing. Botanist, who was the botanist and who was the zoologist? It's not a point of discussion because they don't ask scientists names. They ask beautiful things, and those beautiful things are here. First of all, the first point they have got was this: all living organisms are made up of cell. All living organisms, all living, those who can breathe, okay, who can breathe, that are made up of cell. And then second is cell is a structural and functional unit. This was the second point. Third point, cell does metabolism. You guys, we will study about metabolism more in detail in exam chapter. Yes, we have got catabolism and anabolism. As if you have got an idea, don't worry about, we have got in biology molecules, so don't worry about this thing. So second point was cell does metabolism. This is how it does cellular respiration and it generates ATP. Okay, you guys know this thing. And cell has got genetic material, you know, DNA and RNA. So fourth thing, cell divide. Okay, cell divide and it forms new cells. That is going to be your mitosis, meiosis, cell division. Okay, mitosis plus meiosis, we will cover it in very detail, so don't worry about this thing either. And they call it as omnicellular e-cellular. Okay, omnicellular e-cellular, they call this phrase. So cell divide from pre-existing cell divides and new cell forms. So now there were drawbacks of this theory. This is why it doesn't become law. So there were drawbacks. Its first cell, they didn't tell us what was first cell origin. They didn't tell us how first cell made. They didn't tell us. Then viruses, viroids, and prions. They didn't talk about viroids, and prions. They didn't talk about them. Then they didn't talk about wood cells. Okay, xylem cells. xylem cells, wood cells. You can like thank your god or whatever. I don't know if you love botany or not, but in IMAT, botany is not included. Botany, botany. Okay, I don't remember spelling. Botany is not included. Okay, you don't have to do botany. So that's why wood cells and xylem cells, they didn't talk about wood cells and xylem cells. Now RBCs don't divide. They didn't tell us why RBC don't divide. And first of all, tell me why RBC don't divide, doesn't divide. Why RBC does not divide? Hurry up. Why RBC does not divide? Are you there? Yeah, but for but for division, nucleus is not like a good prominent thing. Okay, okay. If you okay, you are like talking about this is a, like I have got a more critical point for you. You know about neurons, okay? Neurons, okay? We will talk about RBCs later. But you have got neurons. Why neurons don't divide? Why neurons don't doesn't divide? Neurons, the brain cells. Why they doesn't divide? Like the killer people, who killer? Like Iron. And second one, Julia. What about Malaika? What about Alicia? Why? Okay. What about the second, the important person that was here? That was where you? Lohita. Lohita. What about you? Why neurons doesn't divide? You said RBC doesn't divide because they don't have got nucleus. Why neurons does not divide? They have got nucleus. Why neurons does not divide? Don't search it up. Don't search it up. Yeah, Alicia, you search it up and you made it wrong. This statement is wrong. Okay, you talk about. Remember this. They don't have centrioles. RBCs doesn't have centrioles. These second one, okay, it's not about chromosomes. Okay, a nucleus is not a guarantee. Centrioles are the main reason for division. Remember this, because they are going to form the spindle fibers. They are going to divide the nucleus, and RBC does not have centrioles because they don't have two nucleus. Sorry, organelles, and the because mature RBC. Okay, remember this, mature RBCs does not have organelles. They don't have mitochondria, they don't have nucleus, they don't have centrioles. Okay, remember this thing. They just have got hemoglobin, and hemoglobin has got the major function that we will talk about, respiratory system. So don't worry about this. Is very critical point. Remember this: RBC does not divide, neurons does not divide because they don't have centrioles in them. So now I hope you guys got it. That was photo value. And one more important thing, we guys, what is happening? What is happening? So sorry, sorry. I have got like water on my sort of. Yeah, so just wait a second. So what is happening? Okay, now you can see it. Yeah, they don't have centrioles as well, Rania. Their plasma. Now what is it? The what is that material? I forgot that. A layer of plasma. What is called as plasmodesma? There is a special Phragmoplast, yeah, phragmoplast. So like you got me. The phragmoplast forms and they divide. Okay, they divide by phragmoplast formation occurs, and then their material get doubles and they get divided. I hope you guys got it. That is okay. Sorry for that. Now let's talk about structure. Okay, structure of cell. That is going to be super there for you to understand this thing. First of all, I told you cell has got plasma membrane. Then it has got cytoplasm, and in cytoplasm, we have got all the organelles. Okay, in cytoplasm, we have got all the organelles in it, and then there will be a big organelle that we talk about nucleus or all this time. Okay, first plasma membrane, because that is the outermost covering. Then inside it, there will be fluid. Okay, 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 Rania, I hope you got it. We have got like so much knowledge. You will like get so much information. So understand this thing. Okay, first of all, let's me draw a cell here. So this is supposed to be your beautiful cell. Okay, this is going to be your beautiful cell. Okay, I'm drawing two cells. Okay, remember this animal, and this is going to be plant cell. So understand this thing. Animal has got this cell membrane. Let me draw it more bigger. Okay, this is supposed to be animal cell. Inside it, there will be fluid that we like draw it as. Okay. Sorry, let me erase it and let me draw from this way. So this is supposed to be your cytoplasm. I don't know if you guys can see it. This is supposed to be your cytoplasm. Okay. So this is supposed to be your cytoplasm, and let's draw another plant cell as well. Oh, sorry. So let's draw plant cell as well. This is supposed to be your plant cell, and it has also got okay, cytoplasm in it. Okay, cytoplasm in it. Although cytoplasm is spreading out, but that doesn't happen. Okay, it's my pen. So cytoplasm, and now important thing that you guys have to understand here, there will be organelles in it. Okay, organelles in it. So let's like make it red. There will be organelles, different organelles. Okay, they will be like vacuoles, mitochondria, all those things. But in plant cell, okay, in plant cell there will be organelles too. Okay, there will be organelles as well. Okay, now the important thing that you guys have to understand is that plant cell has got an extra layer on it themselves. Okay, extra layer. So this extra layer is called as, now like let's name these things. This black layer is called as plasma membrane, cell membrane, that we call as cell membrane. Okay? This black layer is also cell membrane. Now this red membrane in plant cell, okay, this gray one that you see, it's going to be cytoplasm. Okay, cytoplasm, and inside cytoplasm there will be organelles. But now important thing that you guys, and this is also going to be cytoplasm, a bluish cytoplasm. The important thing, the red one that you are seeing, it is going to be cell wall. So it, remember this, in plant cell we have got extra layer that is cell wall. Okay, and all these red one are organelles. Okay, organelles. We will talk about what are organelles and where are they present. So these are supposed to be all the organelles. I hope you guys got it. I was just trying to let you know what is cell wall and what is plasma membrane. So now you know it. Let's go back to cell wall and do it. Okay, so come here. First organelle, that is going to be our first organelle. We will in the end we will talk about, you will have to do these things, prokaryote versus eukaryote. This is just a single read. I have drawn everything here, so you will just be drawing this. Okay, you will just draw this thing. You will just give it a read and you will understand everything. I will also be reading with you, so don't worry. Now let's talk about first of all cell wall. Cell wall is specific to plant cell, and there are also some organisms that it has also present. So just understand this thing firstly. Okay, firstly understand this thing. The important thing that you guys must know, cell wall is the extracellular in plant cells. Okay, outermost covering in plant cell. We will be very hurriedly. Okay, you have to be attentive so that you can catch up, because we don't have time. We have to cover a very big chapter. It will be absent in animals because animals do locomotion. Plants are steady, that's why they have got the cell wall. It is secreted by protoplasm. Cell wall is secreted by protoplasm. Now tell me, what is protoplasm? Hurry up. What is protoplasm? What is protoplasm? I know. What Malaika, Isha, what is protoplasm? Cytoplasm and nucleus? It's not cytoplasm and nucleus. It's cytoplasm and nucleoplasm. Cytoplasm is protoplasm is cytoplasm plus nucleoplasm. All the plasm, okay, fluids. No, cell without membrane. It's not that. Protoplasm is cytoplasm plus nucleoplasm. Both consecutively called as protoplasm. So cell wall will be secreted by cytoplasm plus nucleoplasm. Okay. So now understand this thing. A plant cell wall has got three layers. Okay, three layers. First is going to be primary cell wall. Okay, primary cell wall. Second is going to be secondary cell wall, and third is going to be middle lamella. Okay, middle lamella. Although it is like a little bit, the picture is not actually, but understand it. I am like speaking it. Okay, primary cell membrane, then primary cell wall, then secondary cell wall, and then there is middle lamella. Now understand first of all, this is going to be the black thing that you seeing. Okay, this black one that I'm drawing black. Okay, that you are seeing. This is going to be one plant cell. Okay, and this is going to be second plant cell. The, that I'm trying with black pen here. So these are going to be two plant cells. They are joined. Okay, they are joined with this green thing. Okay, this green one. Okay, this green one. Now this green one is known as middle lamella. Okay, middle lamella. Middle lamella is just like a sticky fluid that is going to attach both of secondary cell walls of the two plant cells. Okay, two plant cells. Now the second important thing, that these two plant cells will be these plant cells will be circulated by this red membrane. Okay, this red one. This sorry. Oh, sorry. So this red one. So this red one is going to be known as, okay, this is the outermost, and it is known as primary cell wall. Okay, primary. Remember this, primary cell wall. I hope you guys got this thing. This is like very easy. Black is secondary. The green one is middle lamella, that is attaching, sticking both of the plant cells. And now important thing that you guys have to know about the characteristics of middle lamella. So it is going to hold two adjacent cells. Okay, middle lamella is in young plants. It is non-lignified. Okay, it is non-lignified. It could be pectin, that is sticky in nature. Pectin is carbohydrate. Okay, polysaccharide. In mature plants, okay, in mature plants, it will be lignified wood that you see. First of all, in wood that you see, very hard brown wood, okay, that is lignified. Okay, that is lignification of middle lamella. Remember this thing, okay, this is lignification of middle lamella. So it will be lignified in wood. In small plants, it will be non-lignified. It will be pectin, so that will be sticky in nature. And in secondary cell wall, it is the thickest. Okay, in mature, it will be in mature plants, and it is will be permanent. It is the thickest, 5 to 10 micrometer, and the middle lamella is 1 micrometer. Okay, 1 micrometer, just 1 micrometer, and thicker in thickness. Cellulose, hemicellulose, lignin, waxes, silica, these will be the composition of secondary cell wall. It provides mechanical support. Okay, it provides mechanical support. Primary cell wall, it is 1 to 3 micrometer. It is single first layer. Okay, first layer. Yes, you see outside, it is the first layer. It is true cell wall. It is present in all. Okay, it will be present in all. The secondary layer may be absent in some plants. The middle lamella can be absent in some, but primary wall will be present in all plants. Okay, cellulose. Its major composition is cellulose. As you know, cellulose is polysaccharide. We will talk about cellulose very detailed, because there are so many questions arise from cellulose. Just like the day after tomorrow, maybe. Okay, cellulose fibers. There are cellulose fiber present that are in crisscross manner. They will be providing strength and flexibility. It is elastic in nature. Elasticity and plasticity. As you guys know, elasticity is the composition of a material that if we apply pressure or force on it, it retains its property. Okay, it retains its property. Plastic is the region when we apply like a certain limit, we apply force and it got break. Okay, it's got break. That point region is known as that is the property that is known as plasticity. Okay, plasticity, when it comes to its original shape, plasticity, it's going to break at certain level, certain point. Okay, that is known as plasticity. So deposition is of pectin substance. In primary wall, there is deposition of pectin substance. I hope you guys got it. They are not going to ask questions from these. Now they are going to ask you questions from these. Prokaryotes. In prokaryotes, what is the composition of cell wall? It is peptidoglycan, or it is. My voice is not clear. Is there anybody facing my voice is not clear? Is there anybody else facing this problem? Is my voice is bad? Hey, what type of it's clear? They are saying clear. Let me put microphone in it. Let me just wait a second. Can you tell me now? Is it clear? Oh yeah, you can enable subtitles here. Okay, there is CC button, then you can enable subtitles. I have added it. Okay, you can enable subtitles. Okay, okay, if it's better, that's good. That's good. So see here. Now let's talk about others. So these are the important things that you guys have to know. Prokaryotes, peptidoglycan in fungi-like and plant-like protists. Fungi-like and plant-like protists, it will be made up of cellulose. Remember this thing. In plants, it is made up of cellulose. In fungi, it will be made up of chitin. You have to remember this as it is. In fungi, the cell wall will be made up of chitin. In plants, it will be cellulose. In fungi-like and plant-like protists, it will be cellulose. And in prokaryotes, it's peptidoglycan, or it is also known as murine. Okay, murine. So I hope you guys got it. The functions are important. So see here, it has got specific, it provides specific shape to cell. It has got toughness and rigidity. Rigidity is hardness, protection and support. It prevents from osmolysis. It helps in apoplastic pathway, imbibition. We will talk about these things. It was supposed to be in botany, so this is not included. We will do talk about this. Okay, don't worry. Cytoplasmic connections. There will be cytoplasmic connections formed via cell wall that is known as plasmodesmata. And these questions has been asked in past paper. It has been asked in past paper. Okay, I will, whenever it will be, I will be writing pp. Okay, pp, so you can understand it. Okay, so now important one, cell membrane. It is very important. It has been asked in every year, so understand this thing. Cell membrane, first living boundary. Cell membrane is first living boundary. Cell wall was non-living. Cell membrane is living boundary, first living boundary. It is thin, delicate, elastic, and flexible. Repairable, it can be repairable. First membrane in animal cell, as I told you, in animal cell, it will be first membrane. In second membrane in plant cell, because the first membrane is going to be the cell wall. So I hope you guys know cell wall is going to be the first membrane. So that's not a point of discussion because we have done. It's around cytoplasm. So cell membrane always encloses cytoplasm. Inside it, it will be cytoplasm. It acts as barrier. How? It controls what comes in and out, because it will help things to come. It will allow whatever the things needed in the cell and what things are maybe excreted out of the cell. If it gets damaged, it will become dead. Okay, if it gets damaged, it will become dead. The death of cell will occur. Oh, sorry, this is from enzymes. I don't know, my brother has added here, so just neglect it. We will discuss this thing, and it is very important. It has been asked in past papers, and so it is important. So we will discuss this. Okay, don't worry. Now, composition of cell membrane. We study this. This is very important. First of all, this has been asked. Okay, this will be asked in exams that in cell membrane, proteins are more, lipids are more, carbs are more. So what most students does, they mark lipids are more. No, proteins are more. Remember this: 60 to 80% there are proteins. Then lipids, there are 20 to 40% lipids. Okay, and carbohydrate just 1%, less than 1%. Okay, less than 1%. Few quantities, and they will be present at conjugate. So understand, proteins will be globular proteins. Globular protein has got which protein level. We will study about in protein in biological molecule. Globular protein has got which protein level? Is it primary, secondary, tertiary, or quaternary? What is the structure of globular proteins? You guys have to answer these things. And one important thing, yeah, yeah, okay, you are writing in a time. It's tertiary. Yeah, it's tertiary. Simran is tertiary. Okay, that's good. Okay, it has got globular. Remember this: globular proteins has got tertiary structure. And let me ask you, we will cover this in like that question. There is like two cells, myoglobin and hemoglobin plus hemoglobin. What is their protein level? Myoglobin has got which protein structure level, and hemoglobin has got which protein structure level? This has been asked in IMAT past papers. That's why I'm asking. Okay, you guys have to answer. Don't worry. Don't worry about, like, you guys have to participate. This will energize me, and I will like bring more things. If you guys stay like sleep, I will be just like giving lecture, then gone, sending past paper quizzes. You will be solving. That's all we will be doing. There will be no fun. Okay, and I want fun, and you guys really need fun, because if without fun, it will be very bad. Yeah, hemoglobin is quaternary. What about myoglobin? Yeah, Simran, you are right. It's quaternary, but can you specify myoglobin is also quaternary? Hemoglobin is also quaternary. Okay, guys, understand this thing. Remember this thing for forever. Myoglobin is always tertiary. Okay, not maybe, it's tertiary. Okay, myoglobin is tertiary, and hemoglobin is quaternary. Remember this thing, because they ask these type of things. Yes, Simran, you are right. Composition, biomolecules. So understand, protein has got 62. Protein is present in 60 to 80%. They are globule proteins. Lipids. There are two types of lipids present. First are phospholipids that are very like in very high amount. They are more. They provide fluidity. Remember this thing. Cholesterol, they are in less quantity. They provide rigidity. Okay, they provide rigidity. Okay, so phospholipids has got more fluidity. Cholesterol has got less. Phospholipids provide fluidity, and they are in more quantity, present in more quantity. Cholesterol are in less quantity. They provide rigidity, like hardness to membrane. Okay. But in IMAT past paper, there was a question. I will share with you. Okay. Cholesterol provides rigidity. It was written. Okay. Cholesterol provides fluidity. This was written, and this was true. It was written in IMAT past paper. Okay. I'm just exactly writing this statement. Yeah, there are proteins that are integral proteins. Just see it here. Okay. Proteins, just these are big proteins that are intrinsic proteins, and there are also going to be the small glycoproteins. Okay, glycoproteins are more, as we talk about antigens are present. So these are more in quantity, and there will be not only intrinsic protein, there will be extrinsic or peripheral proteins that will be present. So that's why protein are in more quantity. Okay, that's not. I'm not saying it's experimented. So this is how it's more than lipids. Okay, I was also believing, if you haven't studied this information, you might be believing that there is more lipids, and you would have been doing that. But remember this: lipids, proteins are more than lipids in cell membrane. These are books lines I have written here. Okay. So, and one important thing: carbohydrates will present in conjugate form. They will form glycoproteins. So carbs will be either attached to proteins or either attached to lipids. With lipids, it form glycolipids and glycoproteins. So tell me, in which organelle, this is known as glycosylation. Glycosylation of proteins and lipids happens where? Conjugate molecules form where? Okay, conjugate molecule forms in which organelle? Tell me, Anand, where it does form? Simran, where does it form? Yeah, it forms in Golgi apparatus. It forms in Golgi apparatus. Remember this. This is past paper, and I have given question in the in the past paper. If you guys have attempted it, so I have given in the past paper. We are going to discuss it. Okay. Yeah, Golgi apparatus. So unit membrane model. This is very important. In 2020, okay, in 2020 and 2012, this structure was as it is. This diagram that you are seeing without labeling, this diagram was exactly like given. So just understand its importance. Just understand the importance. It has been given 2 times, and it has been asked like very many times, that composition of what is fluid mosaic model. And there were statements written: proteins are in the form of sheets, lipid membranes. Lipids are in the form of sheets. What is the function of intrinsic protein? What is the function of extrinsic protein? So guys have to know about these things. Fluid mosaic model, it was given by Singer and Nicholson. So first of all, before moving to fluid mosaic model, understand first the model was given by unit membrane model, that was given by Robertson. And Robertson told that lipids are bilayer. He was correct in that, but he told proteins are continuous sheet. So he was wrong here. So how it got wrong? Understand this thing. He told this is supposed to be the lipid bilayer. Okay, lipid bilayer, and this is supposed to be the protein sheets. Okay, these are protein sheets. So he told this there are going to be protein sheets. So understand this thing, as phospholipid has got two heads, what to call that, two two parts. Okay, two parts. First is, this is going to be hydrophilic. Head is hydrophilic and tail is hydrophobic. Hydrophilic mean water loving, and hydrophobic means water fear. It has been feared from water, feared. Okay, it has been feared from water. They repel water. Okay, so see here, water will float on this, water can pass. So it means that when water molecule will come, the water molecule will pass from the this hydrophilic area, because water is hydrophilic. Water is like water, it's hydrophilic. So it will move from here. But here you got tail, water can move from here. So it means that this model got rejected, because water molecule cannot enter the cell. But we need Golgi apparatus. As you know, cytoplasm is 90% water. So how we got this water? So this model got rejected. The unit membrane, they asked about in past paper, it has been asked about unit membrane model. So now the model that was given in, I think, 1957. I think 1957, this model was given, Singer and Nicholson. They said lipid is in bilayer, proteins are discontinuous. This was like a good thing. They said globes, they are forming the form of globes, which is embedded in the lipid bilayer. So see here, this is lipid bilayer. These are the globes of proteins that are embedded in the like. Yeah, Alicia, I made it wrong. It's 1972. Sorry, thanks for correction. Okay, lipid bilayer, because they don't ask dates. Don't worry about dates. They never asked about dates in biology, so you don't have to worry about dates. So see here, this is supposed to be lipid bilayer, okay, and there are sheets or globes of proteins embedded. So what important thing you guys have to know, the protein that is like all the way from outside to inside, that is known as intrinsic or integral protein. Those proteins which are just outside, okay, which are outside or that are inside, they are known as intrinsic or peripheral, okay. I hope you guys are getting this thing. And there are also small particles of cholesterol are also present. So cholesterols are also present. There is going to be, this is a lipid bilayer, okay. There are two lipid phospholipid layer that is known as lipid bilayer. And this is going to be, okay, this is protein. So on protein there is carbohydrate is attached, so it will known as glycoprotein. Glycoprotein, because it is attached to protein. And this green one, it is attached on lipid, so it will be known as glycolipid. Remember this thing, because I'm telling you, because this diagram was given and they were asking what is the correct diagram, so you have to like come up. And I hope you guys got it. Now important thing, it is asymmetrical, okay. It is asymmetrical. If you cut this, okay, if you cut from here, if you cut it, you will see the both sides will never be same, okay. Both sides will have got different contents, because they're on outside, okay, outside of the surface there is more glycoproteins and glycolipid will be present, but inside there will be no. So that's why they have got different composition. I hope you guys are getting this thing. Now let's talk about functions. Functions are super important. Understand them very clearly. It is semi-permeable membrane, okay. What does semi-permeable membrane means? It is selectively and differentiately permeable, okay. Selectively and differentiately permeable. It is not completely permeable, fully permeable like everything. It doesn't allow. The protein is amphiphilic. Yeah, it is amphiphilic, okay. It is it has got both parts. It has got hydrophilic and hydrophobic part as well. I'm not saying it is Italian ministry thing, because I heard this first time, not from my books, but I heard this thing firstly from the IMAT past papers, okay. So let me check who is messing, okay. Anyways, so, like, try to understand this thing. It is selectively and differentiately permeable. Let's talk about why it is selectively and differentiately permeable, how it is selectively and differentiatly permeable. So, understand this thing. Small and large molecule will be passed selectively, okay? Selectively. So, see here, some molecules that has got like charge, or those are polar molecules or ions, they will be like so selected molecules will be passed out, not all of them, okay. For example, proteins which are soluble, okay, small size will be selected selectively, though that will like those which cell needs. So whatever cell needs, that will be selected, and it will be like coming inside, or that will be coming out, uh, that will be going out. So that's the like important thing about selectively permeable. Now talk about differentially permeable. Differentially permeable means small size particle can freely pass, okay, freely, like water and O2, hydro oxygen gas. As we know, cell needs oxygen for cellular respiration. So it means that oxygen will be coming without any hassle, okay, without any hurdle, it will be coming inside. So that is differentially permeable. It is very small size, so gas will be like diffusing from outside to all the way inside. So see here, and I hope you guys are getting it. And water molecules, so see, water molecules will be coming from inside the integral protein, okay. Yes, there will be proteins present that are known as aquaporins, small pores that are known as aquaporins, from where water will be passing out. So remember about aquaporins, okay. And glucose will also be dissolved in water, and it will like from outside it will come inside, and it will become glycogen reservoir, and it will store there, and glycolysis will happen. So all things happen. So I hope small size molecules like neutral and non-polar gases will be like coming out there. And selectively permeable, as you know about hormones, okay. Hormones will be coming through selectively, by selective, when cell membrane will select them selectively. Now it is like an important point. It's not written, it's not like related to IMAT. They are not going to ask in IMAT, so don't worry about this. To pass out large polypeptide, plasma membrane secretes through like very big polypeptides. They need to come inside. Plasma membrane secretes second messenger, okay. That second messenger contains some specific hormone that is known as adenylate cyclase, okay. Adenylate cyclase. That adenylate cyclase will take that polypeptide and will take it to inside. I hope you guys are getting. It's not like a very big thing. You don't have to remember this thing. Don't remember this. This was past paper thing. It has been asked in, I think it was asked in IMAT or BMAT. I think it has been asked in BMAT. I'm believing it has been asked in DMAT. Okay, so see here, channel. They talk about what is the function of channels and what about the functions of carrier. Okay, so remember this: channel proteins allow free allow. Okay, they allow free molecules. Whatever comes, they like can bring anything from outside to inside. But carrier proteins has got some specific configuration. Okay, they have got some specific configuration. Anything that will fit in those carrier proteins, that will be taken inside or outside. The carrier proteins doesn't like rely on free basis. They don't offer free services. They only offer premium services to specific people. Just remember this thing. Okay, for example, as you know, sodium and a positive uh, sodium potassium pumps. So they can just pump sodium and potassium ions. We will like do them in nervous system when we will study about nervous system. So when we study about nerve impulse, you will study about in detail what does Na positive gears do, what does A positive gear do, and what is sodium potassium pump, what are voltage regulated, what are non-voltage regulated. We will study about detail because we have to learn the polarization and depolarization in very detail, because I'm at past papers are like super crazy. They asked about these things. Now, I hope you guys got these things. Now, simple diffusion. What is simple diffusion? It is going to be passive diffusion. Okay, diffusion has got three types. Diffusion has got three types. That is supposed to be. Let me draw. It's like separately. I have written here. So, diffusion has got three types. Let me change the pen. I am like getting frustrated from red pen. So, diffusion has got like three types. Okay, that is passive. That we also call it as simple, and there is going to be active, that is going to be utilized, that will be utilizing the ADP. Then there is facilitated diffusion, facilitated diffusion. So understand these. First of all, passive. So first, passive diffusion is simple diffusion. That is just simple diffusion. Anything moving from high potential to low potential. High potential, like for example, high potential to low potential is known as simple diffusion. Okay, as you know, there is like one thing has got very huge amount of quantity that will just spread out here. There is little bit more. So this is known as diffusion. From higher potential to low potential, it is just known as diffusion. So for example, used to play perfume in one corner of room, it will spread out to second corner of room. That is simple diffusion. Facilitated diffusion. Facilitated diffusion, it will happen via some proteins. Okay, it will happen via some proteins. Remember this thing. It will happen via some proteins. And then there is osmosis, that will be happen through membrane. Whenever comes the name of osmosis, remember this thing, it is happening via some sort of membrane. Maybe, as you know, plasma membrane, anything coming out or inside of plasma membrane is also known as osmosis, specifically water. Okay, we will study. You have to like give it, just go and give it a read. Okay, just give it a one read, that would be enough, not more than that, because they don't ask these things. But do know about these things. So there will be important thing, that is active transport, the third type. Okay, third type, it is going to be using ATP. Okay, it is going to be using ATP, active transport. For example, if you are moving things from high potential to low potential, but it would be against the gradient. Okay, against the gradient. Okay, against the gradient. So it means that, for example, there one quantity is less. Okay, this one quantity is less, and you are forcefully moving it to higher quantity. You will need energy. Okay, energy in living body. It is, they haven't asked about hypotonic, isotonic, hypertonic. These things haven't mentioned in IMAT syllabus, but you can study them. Upon for the safer side, you can study them. Okay, but so I would not suggest you to like study them. But for safer, on safer side, you can study them. These are not hard. These are just like one, two lines with one diagram. You can understand them. Any book, it has got a very good explanation. So you are moving from low concentration to high concentration against the concentration gradient. You will require ATP, so that would be active transport. Now understand this thing. One thing, if anything is coming inside of the cell, that will be known as endocytosis. If anything is moving out of the cell, or out of the cell, it is known as exocytosis, because cyto means cell. Okay, so endocytosis can be of two types. What? Phagocytosis, if it anything is coming in the form of solid, it will be phagocytosis. If anything is coming in the form of liquid, it will be pinocytosis or pinocytosis, whatever you call it as. I call it as pinocytosis. Okay, liquid is coming inside, the solid molecule is coming inside. Okay, endo mean inside. Remember this thing, inside. So if you haven't studied about macrophages, then you will study about macrophages like a lot in immune system. But let me tell you, macrophages has got pseudopodias. Okay, pseudopodias. So they engulf solid bacteria. Say they engulf solid bacteria. So see here, this is supposed to be a macrophage. Okay, this is supposed to be a macrophage. So what it will do, there is supposed to be this sort of let's call it as. Okay, so let's call this is supposed to be bacteria. Okay, they will engulf this bacteria. Okay, they will engulf this bacteria. They will engulf this bacteria. So they will engulf this bacteria, and this this is known as phagocytosis. So why I'm writing again. So this is known as phagocytosis. I hope you will study about how macrophages do this, why they do this, what is antigen representing site, how they do this. Okay. Does active use carrier protein and facilitate channel proteins specifically, or they can use whichever actually whichever they want. Okay, active transport can use whatever they want, but that must be against concentration gradient. Okay, that must require ATP. Okay, and facilitated diffusion can be happen through carrier proteins, channel protein, but it doesn't require any protein. Okay, sorry, it doesn't require ATP. It requires ATP proteins. Facilitated diffusion requires protein, carrier or channel, but it doesn't require ATP. When it will require ATP, it will be active transport. I hope you guys got it. Now, if liquid is coming inside, absorption in small intestine, as you know, it is pinocytosis, absorption of water. What is going to be exocytosis? Exocytosis, like now you are removing or excreting something out of the body. Uh, Anything that is moving out of the cell is known as exocytosis. There is going to be secretion, as you know, enzymes and hormones. Hormones and enzymes are secreted out of the cell, so they will be making specific cell. As you know about insulin, and what are other glucagon. You will study about those in chemical coordination hormones. Insulin and glucagon, they are like isolate of Langerhans in isolate of Langerhans in pancreas. Glucagon is secreted by alpha cells, okay, alpha cells, and insulin are secreted by beta cells. So these cells secrete them. This is known as exocytosis. And in excretion, urea is being secreted, uric acid is being secreted. This is known as excretion, secreted out of the body in kidneys. You will study about in homeostasis. Now, understand this thing. Cell membrane acts as defense mechanism. We will like talk about. I told you macrophages represent antigens on their body. When they engulf that bacteria, they represent some part on their body, so that in future, if we get any sort of infection that can be recognizable, their body can produce instant antibodies against them. So this membrane also act as defense. Antigens are present on its surface, as you know. Antigens, as if you know, A, B, O blood groups. These are antigens. Okay, these are antigens that will be present on lipid cell membrane. It also acts as receptor. This membrane also acts as receptor. It will receive antibodies. Okay. Carrier protein requires transport configuration changes. Also required who requires energy. The transport configuration. ATPs will provide energy. ATPs will provide energy. Just let me connect my charger with it. That energy, I'll remember this. Whenever we need anything in the body, okay, whenever we need anything in the body, that energy will be provided via active transport. Okay, sorry, ATP. Remember this thing, okay? Okay, remember this thing. Okay, remember this thing. Now let's talk about this thing, exocytosis. I hope you guys got it. No, passive transport does not use it. But in today's question, we will talk about today's question. Don't worry, we will talk about today's question. Don't confuse it with that, because don't confuse it with that. Active transport, sorry, there will be just utilization of energy when there is something is like very high quantity inside the cell, and we really need to move that thing inside. Say, for example, in extracellular space, there is more water, but the cell requires more water as well. But inside cell, there is like a very high amount of water. That water will be moving inside by using ATP. Okay. there is going to be a concentration gradient. Don't worry, we are going to on that question. Your concept will be clear. Don't worry about that. Okay, so don't worry. Okay, I hope you guys got it. So don't worry at all. Just understand this information for right now. We will discuss questions today, just right after this class, within hour. Okay. Other name, it is also known as sarcolemma in muscle cell. Muscle cell, it is known as sarcolemma, neurolemma in neuron. We will study this in muscles and as well as in neuron. We will study this thing. Okay. Cell surface membrane, it is also known as cell surface membrane. It is comprehensively asked in IMAT, because IMAT Pearson books and A-level books, like comprehensively cell surface membrane, it's the same. Okay, it's not very hard thing. Now, important thing, viruses don't have cell membrane. Remember this thing. This is a point. We will do viruses in very, very detail, because virus is a very this chapter. Questions are like this. Questions are like this. Every year there is question from viruses. So we will be doing that. Okay. Oh, sorry, I wasn't using microphone. You didn't tell me. Okay. We will study about viruses in very detail. There is complete chapter on this. I have got HIV cycle for you as well to understand all the viruses. That will be super cool for you to get more information. And we will bring tons of questions from past papers for viruses, because virus is very, very bad thing. Now, this thing, it has been asked occasionally, not occasionally, it has been asked usually. Okay, usually it has been asked in IMAT past paper. Now, the best important thing in 2023, this question has been asked. Which are the double membranes? So let me show you. Let me show you the past paper, so you can see it. Maybe you understand this. I'm lying, because I just happened to teach in the other class. So you see here. Yeah, this is the question. Okay, just see this thing. Where is it? This is the question. So just see this. This is 2023 IMAT past paper. Okay. They have asked us which of the following structures in plant cell are surrounded by double membrane. So see here, all the organelles are given. So this is the 2023 past paper question. Okay, 2023, big. Okay, let's be quick and do it. Okay, let's do it very, because we don't have time. So now understand this thing. A non-membrane. So you have to memorize this. Just get this is a solid gold. Okay, this is gold for you. Okay, remember this is gold for you. You just have to wear it. Okay, wear this gold. This is gold. This is treasure for you. Okay, treasure. So non-membranous ribosome, centriole, cytoskeleton, nucleolus, chromosome. Nucleolus, chromosome are non-membranous. Single membranes are endoplasmic reticulum. Endo ER is also known is short form of endoplasmic reticulum. I'm just wasting my time. It is known as Golgi body. It is Golgi body. It is not known as Golgi body. It is Golgi body. Lysosome, peroxisome, glycosylation. They are single membranous. Nucleus, mitochondria, and plastid. Okay. This is just as it is written in 2023 IMAT paper. 20. They didn't write chloroplast. They written plastids. So see here, plastid, nucleus, mitochondria. They are all double membrane. So you have to memorize them. They're in your head. These are very easy things, not hard things. Okay. So you will have to memorize. Now let's talk about cytoplasm. Now we are studying cytoplasm, and then inside it we will study all the organelles. Cytoplasm, it is matrix of cell. Cyto means cell. Okay, cyto means cell, and plas means matrix. And so matrix of cell is known as cytoplasm. It is location present between cell membrane and nuclear membrane. As I told you, this is supposed to be like a cell. Okay, this is I'm drawing this cell. Okay, this black one is going to be your nucleus. Okay, and this red one is going to be your all the cytoplasm. Okay, this all cytoplasm. So this red is known as cytoplasm. Okay, this all red is known as cytoplasm. I hope you guys got it. This is easy thing. It's not hard. Now let's talk about components of cytoplasm. This is important. So cytoplasm has got cytosol, inclusions, and organelles. So cytosol. Let's talk about cytosol. So the soluble part of cytoplasm is known as cytosol. It is 90% water and 10% salt. Remember this thing. Cytosol is known as the soluble part of cytoplasm. Okay, soluble, which has got water and salts, because salts are dissolvable in water, so that's soluble part. 90% water. Now there will be two regions, colloidal and true sol. Okay, colloidal cytosol has got two regions. One is colloidal, second is true sol. Colloidal are large salts. Okay, sorry, I'm making it a very confusing process. So now see here, cytosol has got two things: 90% water and 10% solids. So salts are of two, there will be colloidal salts that are known as larger salts, and true sol region that will be containing small size salts. Okay, in cytosol there are two regions, that are colloidal region and second is true sol. In colloidal region, as colloids, if you happen to study solution chapter, that is included in chemistry, and there are like tons of questions from there. We will study about molarity and molality. So see, colloidal region is going to contain large size of salts, and true sol region contains small size of salts. Okay, small size of salts will be present through sol region. And colloidal region has got one sol and gel portion. Okay, colloidal region has further two portions, that is sol and gel. In sol, that will be less dense, that will be center. Okay, in cytoplasm, the center region, okay, central region. So in center, this is known as cell. This is known as sol region. Okay, this is known as sol region. And the other region, that is gel, that is more dense, like more jelly like. Gel means jelly like, more dense. It is will be peripheral. Okay, it will be present here on periphery. Okay, there the periphery will be gel like. It is known as gel. I hope you guys are getting this. When you will study by your own, you will get the idea, you will understand it more, because we don't have time. Literally, we have to cover very big chapter today. So now let's talk about components. First was cytosol. Now let's talk about inclusions. Inclusions is insoluble portion of cytoplasm. In what will be insoluble? There could be glycogen and starch will be present. Okay, these are the storage products that we can use them later for energy production. In animals, it will be glycogen. In plants, it will be starch. Okay, remember this thing. In plants, store the carbohydrates in the form of starch. We will talk about in the carbohydrates, but glycogen is stored in the animals. And waste products, these are supposed to be lipids are like supposed to be wasted, okay, as we burn them very like in high quantity. So lipids are also present in the cytoplasm. I hope you guys got it. There is going to be organelles in cytoplasm, and all the organelles, okay, all the organelles will be present. Prokaryote cytoplasm have only gel. This is like supposed to be very good point for you. Prokaryote cell have just got gel. They don't have got like the sol region, where there is less dense region. They have got more jelly-like cytoplasm. Uh, Functions. Function of cytoplasm, as you know, they will there is this site for metabolism. There all the reactions will happening. Glycolysis will happen in the cytoplasm. We will study about in very detail. We will study about where light reaction happens, where dark reactions happen, where, because these things has been asked in IMAT. This we have to cover them. Glycolysis, glycogenolysis, glyconeogenesis, translation, all these things. And we will talk about each and everything in respective chapters. I don't want to explain right now. And cytoplasm to cytoplasm, the pathway, like the movement of substance directly from cytoplasm to through plasmodesmata. Okay, plasmodesmata that we talked about, plasmodesmata. Yeah, this is known as symplastic pathway. Okay, symplastic pathway is the one cell, like this is one cell. Okay, one cell, it has got cytoplasm. Okay, cytoplasm, this is going to be second cell. Okay, let's like make this way. It's like I have made it wrong. So this is supposed to be one cell. Okay, it's supposed to be one cell. This is supposed to be second cell. From direct cytoplasm to cytoplasm, there will be plasmodesmata present. So the movement of substances from directly one cell to cell is symplastic pathway. Okay, this is pathway is known as symplastic pathway. Now this point is important. Cyclosis. This is the active mass movement of organelles. As I told you, this is supposed to be the cell. Okay. Inside it, there will be organelles. The cytoplasm will be present. Okay. Let's draw with black. So let's draw with black. Why I'm drawing with red? Okay. Anyways, that's cool. So this is supposed to be, and then this is going to be your organelles. Okay. So see here. Let's talk about. Let's call it. This is going to be your any organelle. This is going to be your the mitochondria. So after some time, this mitochondria will travel here. Okay, this mitochondria will travel here. This is known as active mass movement, and it is known as cyclosis. Okay, so see here. Now I hope you guys got the cytoplasm. So first organelle that we are going to study is nucleus. That is very important and very big organized. Let's try to cover it up. And if we somehow got like we didn't cover some topics, that you can study it. Okay, you can give it a read, that proper read. That would be like a good thing. Are you guys understanding what I'm trying to like explain? Is it like I'm going with good speed and you are understanding everything, or like we should like change anything? What are your thoughts? Hurry up, we don't have time. Literally, we don't have time. Are you there? Yeah, that's cool. That's cool. Yeah, a little bit appreciated, because then I will be able. There are emojis present. Okay, you can react with emojis. Okay, you can like have this emoji with the sort of subtitles and screen sharing. There is sort of emoji present, so you can react with those emojis. Okay, there are like different emojis. You can laugh. You can like do whatever you like. Okay, so see here, nucleus. Uh, let's talk about nucleus. What is nucleus? It is stain of nucleus. So what I'm trying to just seeing your reaction, and just I'm trying to explain. Okay, nucleus is the brain of cell. Brain of cell. Tell me, what is the powerhouse of cell? Everybody knows. What is the powerhouse of cell? What is the powerhouse of cell? Where is my charger? What is the kitchen of cell? I know. I knew you all know. What is the kitchen of cell? What is the kitchen of cell? Yeah, now silence. I think now got silence. Huh? Nucleus. I don't know how you got the kitchen. No, it's not kitchen. Yeah, you can say it's. Yeah, Golgi. Lohisa, you are right. It's Golgi operators. Golgi operators is the kitchen of cell. Okay, it's going to process all those things. Okay, it will heat up all those things. Ah, okay. Okay. We have to like we are cooking things Golgi operators, not Golgi operators. It doesn't like sound right. So yeah. Ribosome is cooking things like making cutting. For example, ribosome is cutting the meat. Okay, cutting the meat. Now that meat must heat up. Okay, that meat need to heat up so that we can eat it. So it will go to Golgi operators. Golgi operators will heat it up. So that's supposed to be our kitchen. In kitchen we have got our stove. Okay, fire is in kitchen, as you guys know. I hope you guys know. I'm just trying to make laugh or joking. Okay, I'm just trying to make joke. Don't worry. So nucleus. Okay. Anyways, okay, stain of the nucleus get stained. Okay, with it gives some sort of color with fusion stain. Okay, fusion stain is a stain. If we stain, it will be like purplish appearing, so sort of purplish appearance. Okay, so brain of the cell and crayon, it's the other name of the nucleus. An important thing, understand this: nucleus will occur in eukaryotes. It is absent in prokaryotes, because prokaryotes don't have any organelles. So nucleus is supposed to be the organelle. Occurrence, it will be nucleated and anucleated. Like some sort of plants, some there are some cells that are anucleated. They don't have nucleus. Those are supposed to be RBCs, mature RBC and phloem cells. Okay, phloem cells also don't have got RBC. Sorry, nucleus in it. Phloem and RBC. Phloem in plants and RBCs in animals. Okay, animals. We don't have, and they don't have the nucleus in it. Mature RBCs. Okay. Nucleated, they can be mono, they can be bi, they can be multi. So it means that those which are mononucleated mean they have got one nucleus. Egg has got one nucleus. Okay, so and binucleated, hepatocytes. Okay, in livers they are binucleated. Hepatocytes are binucleated. Paramecium is binucleated, they have got two nucleus. Okay, multinucleated, more than two. Okay, more than two. Skeletal muscle cell, remember this cell, skeletal muscle cell has got multinucleated. And out of osteoclast, bone cell, there are three bone cells. Osteoclast, what is the function of osteoclast? Can anybody tell me what is the function of osteoclast? I know you should tell. Firstly, you should tell. Break down material. Oh, that's good. What is the function of uh, which one, which cell like make up? Okay, which cells make up the bone? Okay, osteoblast. Oh, Maria, you are like seems like pretty sharp. Okay, now tell me. No, osteocytes are the living cells. Osteocytes don't make up. Osteocytes are the osteoblasts are going to form. Okay, yeah, cell to blood. That will be osteoclast. Oh yeah, Rania. Rania, you're right. Rania, where are you from? Rania, where are you from? Rania, is it correct? Right. Okay, you're from Iraq. Okay, okay, you're from Iraq because I Okay, that's cool. That's cool. Just you couldn't join yesterday. I was like curious. Have you watched the recording? Must watch the orientation session we did yesterday. I have sent on your email. Haven't you received on your email yesterday at night? Like guys announced in the Discord. Have you? Are you in Discord server? Are you in Discord? Okay. I have announced there. If you don't receive, do just email me. I don't know. Like most of students receive it, and few students just don't receive it. I don't know what's the issue. Like 2 3 students doesn't receive. I don't know what is the problem. Maybe I should be like sending individually, because I just paste email all emails and it like send. But I don't know why you don't receive. Maybe Google is bad. I am also using the premium version of Google Meet and Google Drive. I don't know why they are making this thing. Okay, okay, Rania, you can send me. Okay, that's good. So see here, nucleus. Okay, now let's talk about the introduction of nucleus. It is the heaviest, dense, and prominent organelle in the body, in the cell. Sorry. Okay, its size is 10 micrometer. Shape, it's usually spherical. Okay, spherical. Spherical mean like circle-like. Okay, irregular. Irregular. It is in an irregular shape in the white blood cell. It is also irregular in shape in white blood cell, but rest in all cells it is spherical shape. Now to observe this, we can observe this nucleus during non-dividing stage. That is supposed to be interphase. Okay, non-dividing stage. We can observe nucleus during non-dividing stage. That is interphase. Okay, during division phase we can't observe it. Location, it is present, it is central in animal cell. In animal, it will be present in center. Remember this thing. Okay, in plant cell it will be on periphery, although in adipose tissues it will be on periphery. What is the function of adipose tissue? What is the function? What are adipose cells? Is there anybody know what are adipose cells? Adipose, in the form of store energy, in the form of, yeah, like you guys know. When you don't know, you get like keep quiet, complete silence. Then you know, like the message is like this, like fires. Okay, that's good. Okay, so see here, peripheral adipose tissue stores fats. Okay, fats. It stores fats. Yeah, it stores fats, and they do. Yeah, and it's helping with thermoregulation. That's very very like good point. You said it's helping. And just feel this thing. In female, okay, in female they have more adipose tissues, okay, in skin. That's why they feel more temperature. They like feel more hot. They're like in temperature they receive, they feel like they receive. That's why they are more sweating, because they have got their adipose tissue, more adipose tissue in their body. Okay, I hope you guys are understanding. Skeletal muscle cell. In skeletal muscle, it will be on periphery. In plant cell, it will be on periphery. So in animal cell, it will be in center. In plant cell, it will be the big, large vacuole will be present in the mid. So plant cell nucleus will be like on periphery. Although there is just one nucleus. Why I'm trying to. So nucleus will be on periphery. Okay, on periphery. Okay. There is going to be one more important MCQ that we will talk in reproductive system. Okay, don't worry. Just like related to adipose tissues, we will talk there. So just don't worry, we will do that. Okay, I don't want to like make so many different things. Now this is the summary of whole nucleus, all the components of nucleus. So just understand this. Okay, just try to understand this. I hope you can see it, although there's a little blur, but I will explain. Okay, I will explain. Just see, focus it here. So first of all, nucleus is just like itself a cell. Okay, it has got nucleoplasm. In cell it was cytoplasm, but it's got nucleoplasm. So say, let's start with, first of all, nucleus is double membranous. Okay, it has got double membrane. So see here, this is the first membrane, this is going to be second membrane. Okay, inside the double membrane there is sort of some pores. Okay, some pores in it. These are the pores. That these pores are known as nucleopores. Okay, nucleopores. Due to these pores, the materials or nutritions will be transported inside to outside of the nucleus, from outside to inside of the nucleus. I hope you guys are getting this thing. So first of all, this is going to be the nuclear envelope. Envelope, always remember this. Envelope means double membrane. Okay, envelope, double membrane. So nuclear envelope separates cytoplasm and nucleoplasm. So inside it is nucleoplasm, and outside it is cytoplasm. So this nuclear envelope is separating the cytoplasm to this nucleoplasm. Now, important thing, outer is going to be the there are two layers. So this outer one is going to be attached. So let's write this way. This outer one is going to be attached with the connected with cytoplasm, and the inner one, okay, this inner one will be attached with this nucleoplasm. Nucleoplasm will be inside it. So cytoplasm contain RER and ribosome, and inside there will be nucleoplasm. I hope you guys know these things. Then there will be nucleopores. Those nucleopores are porins. Okay, porins. Porins protein. Protein. This protein is known as porins. Exchange between cytoplasm and nucleoplasm that will be occurring via these pores. Messenger RNA will be being made inside. That will be exported out from these pores. Salts and enzymes will be coming outside to from outside from cytoplasm to in nucleoplasm. Ribosomes will be being assembling inside and will be moving from pores to outside to cytoplasm. Okay, I hope you guys got it. Now, one important thing: concentration of nuclear pores. So in RBCs, there will be less nuclear pores. Okay, in those RBC, immature RBC. Okay, when there is nucleus present. Okay, in mature RBCs, there will be no nucleus. Remember these little words: mature, immature. There will be 3 to 4. And undifferentiated. Okay, like in egg, there will be more than 30,000 pores, nuclear pores present. In egg, there will be more than 30,000. In RBC, there are just 3 to 4. Now let's talk about nucleoplasm. It is just matrix that contains contains colloidal. That will be there will be more large salts present, and sol region will also be there. Okay, enzymes present, coenzymes are present. There will be cofactors are also present. Now I don't want to explain what are enzymes, what are coenzymes, what are cofactors. We will study these in very detail in very detail in cytoplasm. What was it? We will study in very big detail in enzyme chapter, and enzyme chapter is super important because every year that is just very little small chapter, and every year they ask question from it, so we have to study that cofactor. And then there will be enzyme present like DNA polymerase, RNA polymerase. I hope you guys are understanding it. Now the important thing that is present inside, there will be a big spot inside it. Okay, that is dark region, that is known as nucleolus. It is rough and darkly stained. There will be one too many. Okay, remember this. They ask this thing. How many nucleolus will be present? So there can be in one too many. So first of all, what is the structure? There is central region of nucleolus. Okay, the central region, and then there is periphery region. So this periphery region. Okay, let me change the pen so that you can. This central region and this peripheral region. Okay, peripheral region of. So what will be in central region? There will be DNA, RNA, ribosomal factories. Ribosomal factories will be present inside it. RNA and DNA will be present. On periphery, there will be precursor of ribosomal subunits. So here, ribosomal subunits will be assembled. So they will come inside and they will become ribosomes. Okay, as you know, ribosome has got two subunits, smaller subunit, larger subunit. We are going to discuss this. Don't worry about that because this is past paper question I have given it. So see here, this will be present, and then let's talk about DNA, because if you don't know about the organization, how chromosome becomes being made, like histone proteins and DNA, we will study this in detail in molecular genetics. Don't worry about this because that is itself a very good and very a big topic. So we study that there. Chromatin material, understand this. This is genetic heredity material. So there is chromatin and chromosome. What is the difference between chromatin and chromosome? Understand this. Chromatin is loose material. Okay, loose material. It can be visible with electron microscope. It is only visible electron microscope, and it appears only in interphase when DNA is dividing. Okay, when DNA duplication and replication of DNA is occurring, it is in interphase S phase. We call it as S phase. Chromosome are condensed. Okay, condensed, and they can be visible with light microscope. We can see them. Sorry, and they appear in dividing stage. Dividing stage, they chromosome just appear in dividing stage. When cell is rest, when it's not dividing, the genetic material will be in the form of chromatin. Okay, this. So composition of chromosome, that is very important. Remember this thing. There will be 60% histone. Histone are proteins. Okay, histones are protein, and there will be 40% DNA. So it means that if anybody asks you, if you see question, there are more proteins in chromosome or DNA in chromosome. So try blindly mark there will be more protein, 60% protein. So the ratio will be 3 to ratio 2. And important thing, chromatin has got two regions. Okay, there will be heterochromatin and euchromatin. Heterochromatin is highly closed region and it is untranscribable. It can transcribable. Okay, no coding and decoding. No messenger RNA can form from the heterochromatin region. Euchromatin is open and loose region. Okay, and from here it is can be transcribable protein synthesis like messenger RNA form, and it will go to transcription. It is known as transcription. We were studying molecular genetics. Okay, transcription, and then that DNA molecule, messenger RNA molecule will travel all the way outside, and in outside is it exon introns. Exons and introns are the regions of gene. Okay, genes. It is supposed to be gene, the sub region in DNA. Okay, a DNA in DNA, a specific region of DNA is known as gene. You will study about gene. There are exon, E with E. Okay, this A with A exon is a neuron has got that exon from where information flows. So let me like quickly draw it. I know for you. Okay, you are like making a little bit this one like lecture big, but I hope, yeah, because we have got like different session for you for like asking question. Don't worry. So this is going to be the neuron. Okay, this is going to be your axon. Okay, this is going to be your axon, and this is your cell body, and these are dendrites. Okay, dendrites. Now here you go, there will be gene. Okay, in DNA there will be a promoter region. Okay, promoter region, and then there will be little region exons, and there will be terminator region. Okay, so see here it will be I. Let me draw with this pen. It will be I, E, I, E, I, E, E, maybe I. So see here. Okay, it's okay. So in gene, these are exons or the region that are transcribable but no protein forms. But introns are the region that are not transcribable, protein synthesis occur from there. So we will study in very detail and during RNA formation. So during transcription, as you know, post-transcriptional modifications occur. The capping and tailing and their introns will be removed. So we will study them. Okay, don't worry. We will study in molecular genetics. So chromosomes are condensed, appear in dividing phase, can be visible with light microscope, 60% histone and 40% DNA. And histones are, let me write, this is very important. It will be, histones are basically basic amino acids, okay, basic amino acids, just like lysine and arginine. That's why they will be positive. But DNA has got phosphate group, okay, phosphate group. And it is negative. So that's why DNA will be negative and histones will be positive. Remember this. And I told you ratio is 3:2. This is histone and this is DNA. And now structure of chromosome. So this is, see here, it is chromatid. Okay, chromatid. Now understand this thing. You are going to see this. Okay, this is going to be a chromatid. And important thing, this is two regions of. So see here, this is going to be your. Okay, so this white region. Okay, now understand this thing. This chromatid chromosome has got two arms. Okay, two arms. This is just one arm. So see here, what will happen? That is like beautiful thing. So uh, there is going to be this black one. Oh, sorry, I'm trying again. So this black region that you are seeing, a black circle inside it, this is known as primary constriction. This is known as centromere. Remember the centromere. Centriole, centrosome, centromere. These are very complex things, so like don't confuse these things. Centromere, this is known as primary construction is known as centromere. And important thing, when DNA, okay, sorry, there is going to be one protein present. Okay, one, this red one that I have drawn with red one. This is known as kinetochore fibers. Okay, kinetochore fibers. These red ones, you see, are these red ones. Okay, these are kinetochore fibers. And important thing, what will happen? Those spindle fibers that for DNA, for what is it, for division of chromosome that happens when chromosome will appear on metaphase plate, they will be appearing with the help of those spindle fibers that originated from centrioles. Okay, now those spindle fiber does not directly attach to this black region that we call as centromere. they firstly attach with these red fibers. These are kinetochores. They will attach firstly with these, and then those kinetochore fibers will be attached with centromere. I hope you guys are understanding this thing, because I have given you past paper question. Just right now, I have given you past paper question. So this is the happening. So it means that spindle fiber does not attach directly with centromere. Firstly, they attach with kinetochore, then they attach with. So this is important thing that like you guys have to know. Although our time is going to be like 2 hours just right in a moment, and we haven't like discussed so many organelles right now, so let's get up. Okay, so this thing, I hope you guys are understanding this thing. And there will be like secondary construction. Okay, secondary construction also present, this white one, secondary construction in some chromosome. But this, there on the periphery, there will be a junk DNA, that heterochromatin region that we talked about. Okay, junk DNA will be present, that is untranscribed. Okay, so now let's talk about other thing, that is on the base of centromere position. There will be metacentric when the centromere is in center. So see here, the centromere is in center. Some metacentric near to center. So see here, it is just near to center. Like near to center, it is very away. So it must be like here. Okay, here this is known as metacentric. Then they, oh sorry, I haven't drawn for this, was for like acrocentric. I haven't drawn for metacentric. So acrocentric near to end. So see here, it is near to end. Okay, away from the like just about to touch the periphery. Okay, telocentric at the end. So see here, the centromere is present at the end. So you can like study this. These are very easy. You can like review these notes, and it's written on book. Now you have to remember these chromosomes. Remember this, specific to species. Chromosomes are specific to species. It will be in different species. Drosophila, there will be 8 chromosomes. Frog, 26 chromosome. Human, 46 chromosome. Chimpanzee, 48 chromosome. In pigeons, in 80 chromosome. Pea plant, there will be 14 chromosome. In onion, there will be 16. And potato, it will be 18 chromosome, 48 chromosome. Okay, 48. And if you guys know, okay, if you guys know, there will be, if 2 strands of chromosome present, that will be diploid 2n. And if it happens to be 1 strand, okay, when it happens in meiosis, okay, meiosis, 1 strand remains, so we call it as haploid, n number. I hope you guys know these things. We will talk about these things in genetics and more in cell division, so don't worry. Now I hope you guys know this thing. Let's talk about endoplasmic reticulum. That's going to be a little bit tough, but understand this thing. Okay, endoplasmic reticulum. Uh, so endo means inside, plasmic means cytoplasm, reticulum means network. So it is just a network of interconnected channels. Okay, network of interconnected channels. Just see here. This is supposed to be the this circle is known as nucleus. Okay, so this is going to be the network of interconnected channels. This is known as endoplasmic reticulum. I hope you guys know. Now there can be 3 morphological forms of these channels. Okay, interconnected channels. Okay, for what is it? For endoplasmic reticulum. Okay, I just forgot this because I am not stop speaking from previous 5 hours, and I don't have water in it. Okay, so I am just like feeling some little bit frustrated. So see here, morphological forms. There could be cisternal form, there can be tubular form, and vesicular form. And cisternal form is going to be those that are parallelly arranged. Okay, flattened sacs, they would be just like sac-like appearance, sac-like appearance, and they would be lamellar form. Okay, lamellar form, cisternal, like it is just a morphological structure. Okay, let me draw here. This is going to be a sheet like, so I should draw with another pen. Let's erase it here. What is this angle? So cisternal, they are going to be flattened sac-like. Okay, they will be like little bit sac-like. Okay, little bit sac-like. So these are going to be cisternal. These are going to be the tabular. These are going to be the vesicular. I hope you guys got it. These are these like structure. And tiny tubules away from nucleus. They will be like away from nucleus. And vesicular are tiny vesicles that between endoplasmic reticulum and Golgi apparatus. These are like circles where protein will package and that will go to Golgi apparatus. So don't worry, I will like tell you types. So there's just about 2 types: rough endoplasmic reticulum, smooth endoplasmic reticulum. Smooth, okay, smooth one and rough one. So see here, rough endoplasmic reticulum has got just ribosome. That's why it appears rough. Smooth endoplasm does not have any ribosome. So that's why it appears smooth. Rough endoplasm will be cisternal, morphological cisternal, but smooth will be tubular. It will be more toward nucleus. It will be away from nucleus. It helps in translation. Translation is protein synthesis. As you guys know, we will study in molecular genetics, central dogma of life, transcription and translation. Protein synthesis. If you don't know, transcription. What is transcription? If anybody can tell, what is transcription? What is transcription? Yeah, DNA to RNA. When RNA will form from DNA, it is known as transcription. And what is transcription? Yeah, messenger RNA formation. Yeah, and you are right. RNA to protein. Yeah, RNA to protein. This is known as translation. If you guys know, I hope I just like given you because we will study so far, and in between we will like talk about these things a lot. So that's why I like told you so that you can have like an idea. Those who don't have, although I think majority of you have got, and it's not that hard thing. So see here, one important thing. What does rough endoplasmic reticulum has got? Yeah, okay, then Rania, you are right. So RER has got it is rough. Rough mean it has got ribosome. Ribosome do what is the function of ribosome? It forms protein. Okay, so RER of neuron is also called as Nissl granules. Okay, remember this thing. RER of neuron is called Nissl granules. It provides mechanical support as well, but the main function will be protein formation because it has got ribosome on it. And smooth endoplasmic reticulum will do what? It will do lipid metabolism, cholesterol, also lipids, so testosterone, estrogen, progesterone, all those hormones. These are sex hormones. These are all formed via the endoplasmic reticulum. Smooth endoplasmic reticulum. Carbohydrates metabolism will be happening on smooth endoplasmic reticulum. Detoxification, it helps in detoxification, transportation. The vesicles it will form. Calcium storage, calcium ion storage. In where? Calcium ion storage where? Let me tell. Where does this calcium ion? It will store calcium ion. Smooth endoplasmic reticulum store calcium ion. Where? How? How? Is there anybody know? Is there anybody know how this stores calcium ions in where? Lume. Simran, I couldn't get the lume. What is lume? Is it like a tiny word? In the sad. Guys, you should be like, have you completed your syllabus? I'm just getting curious. It's not psycho. Yeah, Rania, you are right. Okay, in silence, it was right. Sarcoplasmic reticulum. Sarcomeres are different things, Rania. Okay, Rania, sarcomeres are different. We will talk about. Okay, what is botany? I am like you are using. You have studied in Italian, and now you are trying to give test in language. So what is botany? What is it? I didn't see Simran. Bottone. Okay, Bottone. What is it? Button. It's this button. What does it mean? Okay, Simran, it's not that. I think so. See, think this. During in muscles we have got sarcoplasmic reticulum. Okay, whatever symbol you are writing, but it's supposed to be sarcoplasmic reticulum. Okay, I don't know. Calcium storage is in the sarcoplasmic reticulum. As you guys know, sarcoplasmic reticulum is the modified form of smooth endoplasmic reticulum. Okay, Rania, we will talk about what are sarcomeres. Okay, sarcomere is the functional unit of muscle contraction. Okay, functional unit of muscle contraction. It is just a distance between two Z lines. Okay, two Z lines. We will talk about what are Z line, what are M line, what are A band, what are I band, what is milestone, what is action, what blah 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 blah. Okay, don't worry about these things. And this has been okay. Don't worry. There was question from in IMAT 2023. The calcium ion leaks from smooth endoplasmic reticulum. Okay, it is IMAT 2023 question. Okay, so just we will study it. So calcium ion storage. Calcium ion storage in nerve impulse. Calcium ion plays a major role. I know, guys, you don't study here, but just get into med school. Just get into med school. Okay. Okay, so we will study. Okay, Rani, it's just a little bit different. So calcium storage nerve impulse. It helps in nerve impulse. Excitation and de-excitation happens, and there is complete a big block of cardiovascular system. Okay, cardiovascular system, that's very hard, very very hard. Okay, very bad or hard. And that we will study about calcium ions a lot. Okay, lot slow channels, like fast channels. That would be like SA node, how SA node generates like the electrical impulses. You will study that thing. Now you study SA node just generates pulses. That is for you. You will study how that like makes impulses. There will be sodium ions, there will be calcium ions. That's so hard. Okay, just like you are thinking that you just like get into med school, that's going to be very very like hard for you. Don't worry about this thing. You will come to know about how to do ECG. Is there anybody who have done ECG, like who have done like diagnosed get diagnosed with ECG, like a procedure he went through? Anybody went through ECG for heart, like a very sort of. Okay, Ariba. Oh wow, you have done. Oh, you guys have done ECG. Well, do like care about your health. Okay, do run, do jogging every day. Okay, every day do jogging. Okay, those who are fat do every morning, every night. I do running. Although I haven't done like for okay, it's not good, but those who have done, because they are probably diagnosed with sort of a little bit heart problems, that's why they are diagnosed. And most obese person are like have got heart problems, so that's why must like a little bit do sports. Okay, sports, at least do some sort of sport, play such of badminton, or maybe anyway, football is a great, but it's outdoor game, indoor game. Just like badminton, you can play badminton, you can play maybe anything like indoor. Good for your health, very very great for health. So see here, let's move on to Golgi complex. So Golgi complex is very important. See here, it was discovered by Camilo Golgi. We don't know he had been died, so we no need to know about him. But study it because it has been appeared in IMAT. Okay, it has been appeared in IMAT. It had been appeared in. Oh, okay, okay, Ariba, you got this. Because yeah, when we get electric shock, there are just electrical impulses are being generated, so they can like they can mess up. So that's why they recommend ECG, and they do like uh, from there they read your ventricular systole, then the diastole, and they see what is the like impulse rate. All these things are visible from ECG, and ECG also represents your myocardial infarction. Okay, MI. You will be when you get into med school, you will like everywhere hear MI. So MI happens, and angina, heart pain. Okay, heart pain that you see, it is just coronary gets when coronary artery gets blocked. That causes angina. There is like stable angina and unstable angina. So that can be like predicted from ECG. Like medical is very complex. Don't like take it a light. Okay, don't take light. Although that's going to be a very good journey, but like I can like show you like high number of students get drop out. It's not just for demotivation. I'm not saying you have a strong motivation. You will become doctor, very very great doctors, but it's a little bit hard. Okay, just I am trying to let you know, if you are a hardworking student, if you can like study for like very very extra hours, then you should choose this field, and it's going to be very very great for you. Okay, very very great for you. It's very like good field. Those who enjoy studying, they will enjoy becoming doctor, and that's going to be super super very great for you. So see here, Golgi complex, it is also the post office of cell. Okay, post office of cell. It like packages things and gives out thing. Okay, we like did bad thing. It is known as post office. It's not kitchen. It's post office. Okay, it's post office. Kitchen is going to be chloroplast. Okay, sorry. So see here, post office. Those who were telling Golgi apparatus, they told right. So it has got two regions: proximal and distal. Proximal. So see here, if I give you my fingers, you will do whole block. Okay, whole module, a big block about your upper limb. You will study all the muscles, all the veins, all the bones, every like everything, all the muscles in body, like hand. For interior compartment, posterior compartment, then there will be interior compartment. This is cubital fossa. In cubital fossa, which nerve is going, which artery is going, which artery is supplying to this, like this is radius, supposed to be your radius, and this is ulna. So they will ask you, which artery supplies here, which applies to like all the muscles. Then there will be plantaris muscle. This is aponeurosis, hyponeurosis, all these things. Okay, don't worry, just I'm letting you know. Supposed to see, these are my fingers. Okay, these are supposed to be how you can see it. These are my fingers. So this is the proximal region. Okay, this is supposed to be the proximal region of my finger. Okay, and this is the distal. Okay, I'm not flexing. I'm not flexing. I'm not flexing. I am telling you that's going to be a like a big thing for you. You can do it. That's easy, but that's hard, literally hard. Let me tell you. I happen to this was upper, our there was our upper limb and lower limb. Okay, like we have to remember all the vessels, all the muscles, all the nerves, everything. And that happened. I like topped in that module. Okay, I topped in that module, but our 97% of students were fail in that module. So you can like imagine this. Okay, that's why I'm telling you it's like good. It's good because I used Anki flashcards, and if you are like saying I'm flexing, I can show you flashcards. Okay, I that's are like I have made them, and those are like I have to revise them again, and I'm not getting time to revise them. So if those students who use nobody uses Anki in that my college, but I happen to got knew about in my IMAT, so that's why I'm using it. Like I prepared all the upper name, all the lower name, because it's all about memorizing everything. You can like imagine things and you can memorize things. This is how you can memorize. Okay, so this is how I'm memorizing everything. So I will show you. Okay, don't worry, I will show you just in a moment about my math school. Okay, so let's talk about this thing. I hope you guys got this thing. This is the proximal region and this is the distal region. So proximal region is known as cis or forming phase. It is convex. Okay, convex. This is the convex phase, and this is going to be concave phase. It is the inner and maturing phase. Okay, and it is also known as trans phase, trans or distal phase, cis or proximal phase. Now see here, whatever the vesicle will come from, like endoplasmic reticulum, that will get attached here. So it will move all the way from here. It will process inside and it will like get out. It will export it out, or either it will form primary lysosome. Okay, I hope you guys are understanding it. So let me show you my I hope if you might be thinking that I'm sort of flexing, but I'm not. So see here, this is supposed to be all the flashcards that I have got for my the anatomy. Okay, so this is the block B anatomy. It has got like there was some cards are buried, so there are like 5 to 600 cards in it. So see here, we have to study like this. See this thing. Okay, this is extensor dura muscle. So these are the like muscles in it. Okay, so these are supposed to be muscles in it. So you have to memorize all these things. The general function of the muscles of the interior compartment of the forearm is flexion of wrist and fingers. Okay, these wrist are flexing because of, yeah, you talked about flexing. Well, this is the flexion. Okay, this is flexion is happening. I hope you guys are getting this thing. So without any wasting further time, and there is like sort of very big sort of like cards available here that you can see here. Okay, so these are supposed to be all these cards. All these cards you have to like memorize them. I have like made image fluent cards, all those cards. Okay, these are like super great. So I was not flexing. I was like do flexing. It's flexion. Okay, it's flexion. This is flexion. This is extension. Okay. So you will study all about movement. Don't study. It's very good field. Okay, it's very good field, but it's just a little bit hectic. It's a little bit like you will have to work, put more hours. Like other students, you will see engineering students. They will be chilling all the way. Okay, but you will be like working 7-11. So that would be a little bit hard for you. Don't worry, you will do it. Modification. So what is the function of Golgi apparatus? So see here, Golgi bodies. First of all, understand single cisterna. Okay, single cisterna is known as Golgi body. Okay, single cisterna is known as Golgi body. All the cisterna. Okay, all the cisterna. How to draw? I am very bad at drawing. Okay, I am very bad at drawing. I don't know why. So all the cisterna, whatever is present in the Golgi apparatus, these are all known as Golgi apparatus. Golgi complex cisterna plus vesicle with like small vesicles is known as Golgi complex. Okay, I hope you guys are understanding this thing. And important thing, endoplasmic reticulum versus Golgi complex, they don't have ribosome. They are concave plus convex, but endoplasmic reticulums are not. They are not interconnected apparently, so they are not interconnected. Okay, they cisterna are a little bit separate, so not connected to nucleus. Okay, they are not connected to nuclear envelope. On the other hand, endoplasmic reticulum are connected to Golgi apparatus. So they can be cisternal, tubular, and vesicular. All glands, okay, remember this thing. All glands contain higher number of Golgi apparatus. Why? Because they have to secrete hormones, and majority of the hormones are protein in nature. So it means that they require a cell require the like tons of Golgi apparatus for processing of hormones, for processing of those proteins. Now functions, modification of molecules, carbohydrates plus protein, glycoproteins will form here conjugated molecule. Okay, it is known as glycosylation. I told you earlier. Lipids plus proteins, it is known as lipoprotein. Polysaccharides will form in this storage, export, phlegmoplast formation, formation of cell wall that we talked earlier. Okay, it doesn't have cell centrioles, but Golgi apparatus will form phlegmoplast. It will do this formation of cell wall correctly. Okay, acrosome. Acrosome, the sperm head. If you guys know the sperm head, we study in reproductive system in male. So this sperm head is acrosome, and it is called. It has got lysosomal enzyme. So it is like derived from Golgi apparatus. Lysosome, as you know, it has been made from Golgi apparatus. It is secreted by Golgi apparatus. Membrane formation, cellular packaging. Okay, as you know, if there is sort of any membrane, okay, this is a sort of membrane, this Golgi apparatus will secrete, if this is vesicle, it will contain some sort of protein. So this will be need to be exported out. So what will happen? It will like come here, and this vesicle like get this, and this content will empty out. But now this layer of this Golgi, sorry, sort of vessel will become the membrane. Okay, this will become again this. Okay, so membrane will become big. Okay, bigger. Now this is the important thing that has been to ask two times in IMAT past papers, two times it has been revised. Okay, enzyme formation. Then what will happen? Enzyme formation, rough endoplasmic reticulum will then after sorry, enzyme formation will occur on Golgi ribosome. After ribosome, it will go to rough endoplasmic reticulum, from where the sort of vesicle will form. In that vesicle form, it will send to Golgi apparatus for modification. Golgi apparatus will further modify it, and it will secrete in the form of vesicle again. It will known as secretion, and it will be exported out. Okay. So unit of Golgi apparatus is dictyosome in plants. Okay. In plants, the unit of Golgi apparatus is known as dictyosome. Dictyosome, Dictyosome, dictyosome. Now this is the actual problem. Ribosome plus endoplasmic reticulum, then smooth endoplasmic reticulum, then forming phase of Golgi apparatus, then maturing phase of Golgi apparatus, then primary lysosome will form. This is the formation of primary lysosome. Ribosome will form plus endoplasmic reticulum will form the enzymes, those hydrolytic enzymes. So they will go. We will let's talk about first of all lysosome. What is it? Is splitting body. Okay, they can like burst in the cell, and they can like kill everything. So they are spherical sacs. They will originated from trans phase of Golgi apparatus. They contain hydrolytic enzymes. Okay, hydrolytic enzymes are of 40 types. They will be proteases, lipases, phosphatases. Proteases that will be breaking down proteins. Lipases that will be side bags. The lipases will be breaking down lipids. Phosphatases will be like breaking down phosphates. Okay, phosphodiester bonds. So the enzyme synthesized in rough endoplasmic reticulum then sent to Golgi apparatus for modification in the form of vesicles. Okay, after those enzymes will like pack into vesicles, and they will known as lysosome. Lysosome are single membrane organelle. Okay, remember this, and work in acidic pH because those are acidic enzymes. 4.5. They work in acidic enzyme, occur more than usual. They will occur more than usual in which cells? Lymphocytes, neutrophils, osteoclasts, sperm. In sperm, we got loose acrosome has got hyaluronidase that are so that will be present in fluids. Tears has got lysosomes, saliva has got lysosome, sweat has got lysozyme. Okay, lysozyme that is secreted through saliva. It has also got the lysosome in it. Okay, so now understand this thing. One primary lysosome will form from the Golgi apparatus that will contain enzymes. So phagocytosis will happen any foreign particle. Okay, this is bacteria, supposed to be this is bacteria that is coming inside of the cell. So this bacteria came inside, but it is encircled by primary lysosome. Okay, sorry, no. Let me tell you. This was the membrane. Okay, this was this membrane. This primary bacteria or any foreign entity came inside via this membrane. So this is encircled in this membrane. Now primary lysosome will spot this, and it will come and encircle this. Okay, encircle this whole molecule. So primary lysosome will come, and this is primary lysosome. This will encircle all of that this one. Okay, all of this will be engulfed by this primary lysosome, and now this can blast, and inside that bacteria will be like dead. Okay, I hope you guys are getting this thing. These are like sort of like not hard points, easy points, but point of remembering. You have to memorize this thing. Now, primary lysosome, they are inactive and fresh. Secondary lysosome, active lysosome. They are primary lysosome plus food vacuole. That is phagoplast. Okay, sorry, phagosome plus primary lysosome is known as secondary lysosome. Tertiary lysosome, residual plus waste. Okay, when they will burst, those waste and residual, that will be known as tertiary lysosome. And quaternary are autophagy. Autophagy, you know what is autophagy? When there is any dysfunction in the membrane, sorry, in any organelle of the cell, that lysosome will circle that dysfunctioned organelle. Okay, this is the lysosome that has been circled that mitochondria, and now it can burst. Okay, it can burst, and that this is known as autophagy. Okay, autophagy functions. Intercellular digestion from ribosome RER, it will send to a smooth endoplasmic reticulum, then forming phase of Golgi apparatus, then maturing phase of Golgi apparatus, then primary lysosome will form. Autophagy, self-eating, it will recycle. So starvation, I don't know why I have written starvation, but it is self-eating of any organelle or cell. It is known as autophagy. Autolysis, differentiation or morphogenesis, change of any morphological form. Okay, so you have seen that when we are babies, okay, babies in mother belly, okay, in mother womb, we have got fingers. These are connected to each other via skin. Okay, skin. So lysosome are going to separate this. Okay, lysosome are going to separate these fingers. It is known as autolysis. Tail removal, we have got tailbone, although tail doesn't form, but it is believed that tail is being removed via these lysosomes. Then in tadpole, we have got the, in like little frog, the earlier stage of frog, tadpole, it has got tail, so that tail will be removed and mature frog will form. So that is also due to autolysis. Now there is apoptosis, that is known as straddle bags. They will, when there is dysfunction in cell, okay, cell has got like mad or cell has become uh, carcinogenic, cancerous, these all lysosome will like accumulate in the cell and they will bust and that cell will be dead. This is known as apoptosis. Significance. Okay, you can study this thing. I don't want to explain this because it's also not included, but you can like study this. These are 2 diseases. You can study this. Okay, just mark it down for your future read. Vacuole. Vacuole is very important. We will do like vacuole, ribosome, mitochondria, and then plastids. Okay, yeah, hurry up. We can cover them up like very quickly. So see here, vacuole. Vacuoles, specialized vacuoles perform the function of lysosome. Okay, specialized vacuoles perform the function of lysosome. So first of all, understand vacuous means empty. So vacuole is just a spherical sac. Okay, it's just a sac. Now it will be originate. It will originate from Golgi apparatus, endoplasmic reticulum, or plasma membrane. Okay, these are the 3 origins from where vacuole can form. And vacuole has got single membrane. I have written in the previous where I given you a trigger that you have to remember the name. One important thing occurs in animals, there will be small and many vacuoles will be present. In paramecium, there will be contractile vacuole present. Contractile vacuole are also known as food vacuoles. Okay. Plants, in meristematic tissues, it will be absent. In young plants, it will be, these will be small vacuoles present. In mature plant, there will be single large vacuole will present. Okay. And many small vacuole forms large vacuole. So coalcine, okay, coalcine, Coalescen, coalcine. It is a phenomena where many small vacuole form large vacuole. It is known as coalcine. And though this is just a vacuole is not that important. There is function is important. It will store water. Turgidity, when but it will swell with water. It is known as turgid. The phenomenon is known as turgidity. It will protect. Okay, compound will accumulate and protect from organisms like in wood. You see it is very hard, so there will be lignin will I told you lignin, the deposition of lignin. So lignin will store into vacuoles and that will become hard, so it will protect the plant. So wood will be very hard, so it helps in protection. Ribosome is very important. Understand this: 50% RNA, 50% protein. 50% RNA, 50% protein. No DNA. Now understand, Formation in nucleolus region, we talked about the central region, ribosome factories. It will form nucleolus, will transport into cytoplasm. It will form in nucleolus. You can ask where ribosome forms. Okay, you have to ask nucleolus. It will be transported to cytoplasm. It can be exist in two forms. Okay, free form. It will free in the cytoplasm. Free form into cytoplasm. Attached to rough endoplasmic reticulum. It can be attached. It can be free. Structure. It has got large subunit and small subunit. So see, this is the large subunit. Okay, this is the large subunit, and this is the small subunit. Okay, this is small subunit. This is small subunit. This is supposed to be large subunit, and inside it, this like the rope you are seeing, this is messenger RNA. This is translation is happening here. Okay, translation is happening here. So see here, larger subunit and smaller subunit needs Mg positive ion for their attachment. Okay, Mg positive ion. This is how translation will happen. Remember this, they can ask you which ion is needed for translation, for attachment of larger subunit, smaller subunit, magnesium ion, magnesium ion. Remember this thing. This is very, very important. Like important. Okay. So polysome, many ribosomes on messenger RNA. So see here, many ribosomes are attached on messenger RNA. It is known as polysome. This structure is known as polysome. How many proteins will form from polysome? Protein is equal to number of ribosome attached. So the more the ribosome, the more will be protein will form. It's not like specific thing, but important thing that is in prokaryote cell it will be 70s. In eukaryote cell it will be 80s. And now the important point is that my charger got disconnected. I don't know what's the bad thing is happening here. I am just very uncomfortable sitting here. So sorry for that. So sorry for that. Uh, so see here, what I was telling you, this prokaryote has got 70s. 70s S is just a sedimentation rate. Okay, sedimentation rate. So you know, centrifuge is a machine where we like spin different materials and they separate out on their sediment rate. So 50s and 30s, one larger subunit will be 50s. Sedimentation rate of larger subunit 50s and smaller subunit is 30s. But when they are both combined, their sedimentation rate will be 70s, not 80s, 70s. Remember this thing. Okay, you don't have to calculate these. This is just their individual sedimentation rate. And in eukaryote it will be 60s and 40s, and the overall sedimentation rate will be 80s. Okay, mitochondria, that is super important. And it is endosymbiotic organelle. Which two organelles are endosymbiotic organelles? They can ask which two organelles are endosymbiotic. Which two organelles are endosymbiotic origin? Has got endosymbiotic origin. Chloroplast and mito. Yeah, you can write it very like little. Mito and chloro. Yeah, that's good. Mitochondria and chloroplast. We will like talk about this mitochondria. So mito is mean thread. Chondrion mean granules. Occurrence, it occurs in aerobic eukaryotes because it does aerobic respiration, due to which ATP is formed. We will be doing it in bioenergetics. Bioenergetics. Do join that session. That's going to be like we will do quickly and we will do it. Okay. Significance, it helps in cellular respiration. Shape, it has got round oval shape. Okay. In sperm, it is in spiral shape. In sperm, it is spiral shape. Presence and number. So see here, it is absent in RBCs. Mature RBC, it will be absent. It will be present in less amount in adipose tissue. It will be more in muscles. Okay, muscles, because we are exercising, so we need more ATPs. So more mitochondria will be present. Now see here the structure of mitochondria. So it is double membrane. First of all, it is double membrane is okay. Now understand first of all this thing, the outer membrane and inner membrane. So this is outer membrane. This is going to be smooth and it will have porings, those small pores. Okay. Inner membrane will have infoldings that are known as cristae. So you are seeing these are cristae. Okay, this is inner membrane. It has got in cristae. You don't have to confuse cristae with cisternae. Just remember cristae is in mitochondria. Rest all are cisternae. Like in endoplasmic reticulum, this will be cisternae, and this will be in Golgi apparatus, that will be cisternae. Okay, I hope you guys understand. Now understand, just like nucleoplasm, cytoplasm, mitochondria has got this new matrix. Okay, matrix that is just like cytoplasm. Now what will be in mitochondrial matrix? There will be DNA present. That will be circular DNA, just like prokaryotes. Okay, prokaryote has circular DNA. And earlier endosymbiotic theory, we are going to talk about endosymbiotic theory. So it will have DNA that will be circular. That's why it is self-replicating organelle. This has been asked in past paper. RNA is going to be what RNA is going to be present in the nucleoplasm, matrix of it. Ribosome 70 years, prokaryotes ribosome, okay, will be present here. Then there will be, what will be present here, okay. This is enzymes and cofactor and coenzymes will be present, okay. Enzymes, coenzymes and cofactor will be present, as we are going to talk about succinic acid dehydrogenase. So those are enzymes and different, are there will be different enzymes. We will talk about what are the enzymes in bioenergetics, so don't worry about these things. I hope you guys got it. Now, what is going to present more, how the ADP will be forming. So see here, on Christie, okay, on Christie, there will be, this is supposed to be your Christie, okay, this is the Christie. So this is supposed to be Christie. On Christie, Christie is also known as oxysome, okay, oxysome. Oxysome. So Christie has got elementary part or fundamental particles that are known as F naught, okay, F naught or F1 particle, okay. This is just the zoom variant, okay. This is the elementary particle F1 F naught particle. This is the like zoom variant of F naught particle. So see here, the base is known as F naught and the stark is known as F1. So ATP synthesis will be happening on, okay, ATP synthesis will be happening on these F1 F naught particle. And important thing, in Christie, on Christie, there will be oxidative phosphorylation will be happening. Is there anybody know what is oxidative phosphorylation and what is photophosphorylation? We will talk about in bioenergetics. I'm just asking for like, I just want to like flex. Just which person has asked? Which has asked? Okay, Divyash. Is it Divish? What his name has been pronounced? I don't know. Divyash, 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 Divyash. Is it right? Divyash or Divyash? Okay, Divyash, Divyash, Divyash. Okay, Divyash. So now Divyash. Say, is it right, Divyash? I will call you just Div. Okay, I don't call it Divyash or whatever it is name. I will just call you Div. Just understand it. Okay, you are just Div here. So I will just call you Div. Okay. And now tell me, what is oxidative phosphorylation and what is photophosphorylation? Silence in the house. Hurry up, guys. We don't have time, literally. Hello, guys. Oxidative in respiration. What is oxidative phosphorylation? Oxidative in respiration. I know these things, but what is oxidative phosphorylation? Oh my god, yeah, it is synthesis of ATP in presence of oxygen. And what is photophosphorylation? Yeah, that's correct. Synthesis of ATP in presence of oxygen. And what is photophosphorylation? What? Maria, what? Are you right, brother? Bro, ADP to ATP is not formation. So then to ATP, yeah, it says formation of ATP in the presence of sunlight is known as photophosphorylation. Yeah, yeah, Rana, you are right. Rana, you have got like good concept. So you are right. Okay, you are right. Okay, that was thing. Now let's talk about oxidative phosphorylation and photophosphorylation. We will talk about in detail in bioenergetics. So you will like absorb content. Okay, I mean this is what I told you yesterday that this will be your, yeah. If you said ADP to ATP, that's not. That is just phosphorylation. Okay, that is just phosphorylation. Yeah. Okay, let's talk about plastids. Plastids is going to be very important, and let's talk about endosymbiotic theory first. So I am going back into the very like evolutionary mode, 3.5, 1.5 billion years back. Okay, so guys, once upon a time there was one prokaryote. That prokaryote was just prokaryote. Okay, it was little prokaryote that can use, okay, that can use, okay, are you guys understanding? I'm just into, I don't have costumes, otherwise I would have wear like a sort of white clothes and big gray hairs so that I could have gone into 1.5 billion years back. So see here, there was just one. No, at that time human doesn't existed because Aerobic, there was no there was no mitochondria. The mitochondria is going to farm right now. How that could have that could have been there? How I could have been human? So I'm just telling a tale, okay? That procreate was procreate. Okay. So see here, this was a procreate that can utilize oxygen. Okay, that could utilize oxygen and can produce ATP. But that procreate has no shelter. This procreate does not have any house, so it needed house. But there was another procreate, okay, big procreate that has house, like a big place, but it don't have energy. Okay, it needed energy. So they said, "Hey brother, let's collaborate. Okay, let's collaborate and let's become buddies. Okay, I will give you shelter. You should give me energy." They agreed, like they set the agreement, and this agreement is going on for forever. Okay, so it has give it get into this procreate. Okay, and this is why earlier it was single membrane, but the second membrane it got from here when it got into phagocytosis. Okay, this membrane, the second membrane, it got from here. Okay, this second membrane, I hope you guys getting it. So that's why the mitochondria is double membrane. So it got in, and this is the endosymbiotic theory and endosymbiosis. This is endosymbiosis theory, and two organelles, mitochondria and chloroplast. These two organelles are organized originated from this endosymbiotic theory, endosymbiotic relationship. Okay, plastids. First of all, let's talk about plastids. Plastids occur in plants and algae cells. Okay, what you call is algae, whatever you call it as, it's I call it as algae, and now I'm calling it as algae, algal cells. So proplastids, there's immature plastids. Okay, now let's talk about proplastids. So proplastids are chloroplast, chromoplast, and leucoplast. Okay, three types. Where is it, brother? Where is it, brother? Brother. Chloroplast, chromoplast, okay, chromoplast, leucoplast. These are three types of plastids. Okay, three types. So chloroplast, chromoplast, leucoplast. Let's study first chloroplast, then we will cover chromoplast and leucoplast. So what is chloroplast? It is endosymbiotic organelle, just like this one. Okay, it organized from here this way. And understand, it is green in color. It is present in mesophyll cells. Okay, mesophyll cells has got 20 to 100 chloroplast. Mesophyll cells are just leaf cells. Okay, leaf cells. You see, okay, on leaves. Sorry, what I'm saying. Mesophyll cell has got 20 to 100 chloroplast. And what are proplastids? Proplastids are just immature plastids. Okay, they are plastids are of two types. Okay, plastids are of two types. One is proplastid, and second one I haven't written here because that's not included in our book, but it is known as something. You can search about it. Okay. So first of all, let's talk about, don't study about, just take this as plastids are of three types: chloroplast, chromoplast, and leucoplast. So let's talk about chloroplast. Chloroplast has going to be green color. Mesophyll cells are present in leaves. Okay, leaves of plants. There will be 20 to 100 chloroplasts. So G, this is the chloroplast. It is also double membranous. Okay, double membranous. But understand this. This is the outer envelope. Okay, outer membrane and inner membrane. This is known as envelope, outer membrane and inner membrane. Inside it, there will be this matrix, just like in mitochondria it was matrix. In the chloroplast, it is known as stroma. It contains DNA. Circular DNA will be present, just like because it was prokaryote. Self-replication will occur. RNA will be present. Enzymes, coenzyme will be present. Ribosomes, 70 years. Okay, and protein that will be present. Enzyme, that is Ribosome, and Ribosome is the most abundant protein in the universe. Okay, Ribosome is most abundant protein in the universe, and we will talk about Ribosome in bioenergetics. So don't worry. It is semi-autonomous as well. Okay, semi-autonomous. It means that it uh, it does what? What does it do? Semi-autonomous means, what does it mean? Semi-autonomous. It just like name reflects. It can make proteins for itself. It has got its own DNA, so it is semi-autonomous, but it is dependent on glucose. It don't have glucose. Sorry, it doesn't. It doesn't have like what things are it needs. It needs photosynthesis for photosynthesis CO2. It doesn't have CO2, so it depends on whole plant cell. Okay, this plant cell to give him the sort of that CO2, so it can form glucose inside it. So it is semi-autonomous. It can like half functions it can do by itself, but half functions are dependent on other cells. So one important thing in stroma, dark reaction will happen. Carbon fixation, reduction, regeneration. These are the three steps of dark reaction that will happen in stroma. Then there is grana. Okay, grana are the piles of stacks, and this one stack is known as thylakoid. Okay, this one stack is known as thylakoid. Remember this. This one stack is known as thylakoid, and in the membrane of thylakoid, light reaction will happen. So granum is known as this pile. Okay, this whole pile is known as granum. Pile of thylakoids. Thylakoid is flattened sac. Thylakoid membrane got pigments. Light will capture Z scheme. Z scheme is known as light reaction. Light reaction. Okay. So in lumen of this sac, photolysis, H positive photolysis will happen. Breakdown of water. Okay, Rania, yeah, I got it. It can divide into its own without getting the whole cell into division. And it can also make its own protein. Yeah, yeah, you are right, Rania. What is it? You are right, Rania. We will come up to this. I'm just very bad at names, so I just like well, I don't want to pronounce names differently so that it looks very bad to the person whom I'm like narrating. So photolysis will happen in the lumen of thylakoid, and H positive ions accumulation will be happening in lumen. We will study these things in detail in bioenergetics. Don't worry about this. These are all reactions in there, and I hope you guys got it. So this was just chloroplast. You will going to study this from book. This will make all these things will make sense. Functions. It is green in color. Photosynthesis will happen. Storage of starch will happen. Stromal synthesis ATP. Okay. In stroma, synthesis of ATP happen. We will study how. Okay, Rania, I'm saying right. That's good. Evolution of O2. Okay. Evolution of O2. Photolysis. We will study about photolysis, how it happens. Everything in bioenergetics. We don't have to study it right now. So this was just cell biology. Now you guys are going to study. So we have covered mito, Golgi, endoplasmic reticulum, nucleus, plastids, ribosome, lysosome, vacuole. You are going to study this by own, self-study it. Okay. And now animal versus plant cell, I have drawn here. Okay, animal versus plant cell, and the second one is prokaryotes versus eukaryotes. You are going to study from these notes. These are present in Discord. Just click the link, go to website, and download from here the cell biology notes. Okay, cell biology notes. So here you go, and you will find them this prokaryotes versus eukaryotes. So it means that in prokaryotes we have got flagella, which is made up of flagellum. In eukaryotes, it is made up of tubulin. Nuclear envelope, it does not have. It have nucleus. Prokaryotes does not have. It have nucleus. So all these things are written like tons and tons of things, like very very detailed. I have written everything that I can. I think I had wasted like one day whole on this thing. Like searching up and writing everything, so you can like go here and write everything from here. Yeah, like study it. Just give it a read. And eukaryotic cell. So see here, I have written plants versus animal cells. So you can see cell wall present, not present. So I have written everything. You can like study it. It's not hard. I have I have used the Pearson Global Approach 11th edition. So, but I used like tons of lectures. I've just used so many things I can like say to you. But majority book was Pearson Global Approach 11th edition. I don't recommend up to you all. Okay, those who don't have books, those who just want to follow your national book. Additional, you can follow if you like want to follow Cambridge A level book. Just read it from Cambridge A level book. Simran, you are going to read Cambridge A level book. Okay, I am going to share the PDFs. If you can buy the hard covers or hard forms, you can like buy the physical books that will be available on Amazon, or yeah, you can buy from there. But I will share the PDF that you can download on your laptops, tabs, iPads, whatever you are using, and you can start reading. I hope you guys got these things. These things. So three things are going to be your reading. So you are going to read these three things. That will be like these are very well very detailed. So you are going to study cytoskeleton, animal versus plant. Everything is written. You can study this from book. Okay, any book. Animal versus plant cell on from my notes, and then additionally you can study from book. I hope that like lecture was good. What was your like? What is your overview? You can give me a feedback. Is there anything we can change? Although we can reduce speed. Okay, don't talk about speed. We can reduce speed. We will be like more speedy. Although we were like less speed. I was like teaching very less speed. We will be more speeding up because we have got so many things to cover, and you will be doing tons of self study as well. We are talking about self study. I have written here, so we are talking about that. So how was it? Have you understood? Like, was it like good? Okay, thank you. Okay, okay, okay. Thank you, everybody. Okay, I that was like a good. Now it gives me motivation. And tomorrow we will be like with more good energy. Although tomorrow is going to be chemistry, I will share a schedule. Okay, okay. Thank you, Alicia. Thank you, Mr. Everybody, Julia. Anand, thank you. Thank you. Yeah, I have to speed it up. They will speeding up because we have so much on the plate. That is physics and logical reasoning. Biology is so big. We have just drawn here. You can see it, like the table of contents. I will share the pictures in as well in this Discord. Okay, I will share in the Discord. You can utilize them, but you can see this. This is like very lot. I will share in the resource section. Okay. Now let's go back to our questions. Okay, past paper questions, because past paper questions are supposed to be good ones. So let me open 14 students solve them, and nobody got full marks. Nobody got full marks. That's like not a good thing, but a good thing. Okay, okay, right. Yeah, that's bad. Out of 6, many students have got 2 marks, like 4 options wrong. These were past papers, so just like get the idea that they are going to ask you question like this. I hope you guys are getting this. I can add like feedback for you. I hope I will be adding feedback. You guys have to be show up every day. If you didn't show up every day, that would be like a bad one for you. Okay, so let's talk about different things. Okay, we can. What was the next name? Somebody, yeah, maximum. There will be no chapter wise test. We will not be doing chapter wise test or mock exam. We will be doing complete weekly mock exam. I have showed you. If those who haven't watched the orientation session, I am just, my mouth is so dry. Okay, we did orientation session yesterday, and we talked about at the end of the week you will have mock exam. You will be attempting that. Okay, so I'm just sharing my screen right now, and let's do it. So I hope you guys can see it. So this is going to be our first question. Okay, can you see my screen? Is it visible, the question screen? Yeah, yeah, we can see it. Okay, okay, if you can see, that's cool. Let's talk about question. The first question, the diagram below represents a fluid mosaic model of the cell surface membrane. Now see here, it is the same model that I told you. What is this? This big one, integral protein. This is phospholipids bilayer. This is going to be our intrinsic protein. Okay, intrinsic protein. Okay, and this is going to be our what was it? Cholesterol. This is going to be our uh, glycolipid. Sorry, glycolipid. Okay, glycolipid. I hope you guys are. So only 2 of the labeled molecules have both hydrophobic and hydrophilic areas. Which 2 molecules are they? So we know about phospholipids, and we don't know about any other. So we have to come up. And I didn't know. Literally, I didn't know earlier, but I happen to know from solving the figure when I did this past paper at my time. Then I got to know about integral protein also have got the hydrophilic and hydrophobic areas. I didn't know earlier. Did you guys know? Did you guys know this about this uh, integral protein? Because I didn't know. Literally, I didn't. Okay, if you know, that was like cool. But integral protein also has got, so this option is right. It's not. It's just MCQs about memorization. So if you have got, I told you yesterday that it's all about learning concepts and then memorize them. Those students who are going to memorize more than like all others, they are going to perform well. So this is the case. So now let's move on to second question. So see here, what is it? Which one of the following is not correct about human chromosome? They contain regions called genes. Yeah, we know that they contain genes. This is true. So solving question is just highlight which is not correct. Okay, not correct. They contain regions called genes. Yeah, they contain regions that are called genes. So it is true. They are made of DNA and protein. Yeah, this is true. Histone and DNA. We have talked. They are sometimes not found in pairs. Okay, they are not sometimes found in pairs. Yeah, they cannot. We talked about haploids. Okay, Haploid we did talk about. So C is also true. D, they can attach to the spindle at the centriole. No, brother, they can't attach the spindle at the centriole. They attach spindle at the centromere. We talked about centriole is an organelle. So see here, somebody put C, but option D was like that one. So I hope you guys are understanding. So E, they are sometimes found in pairs. Yeah, this is also true. E option is not correct. Okay. Which one of the following transport mechanism? Which one of the following transport mechanism require the use of protein molecules found in membranes and ATP? Now understand ATP. First of all, understand diffusion does not require ATP. Facilitated diffusion does not require ATP. So these options to get straightforwardly minus. There was like this was an easy question. I know, Rania, you are saying about membrane transport. The one required both ATP and protein was only active transport, not both active. Yeah, I have written active transport only, right? Yeah, see here, it is active transport. I don't know what you put here. So see here, the D option is right. Only one. Active transport is right. Diffusion and facilitated diffusion does not use proteins. It can happen like anywhere. Facilitated diffusion does use proteins, but it does not use ATP. So it option gets like minus because we are required that ATP is also being used. So active transport is the only option that we left with. I hope you guys are getting this question is like now you can do it. So I hope whoever like made this wrong, now you got the answer because we talked about it's going to where protein will form. Firstly, ribosome, then it will package into the vessels of endoplasmic reticulum, then it will be transported to Golgi apparatus. I hope so. This which one option is the right? So see here first is going to be ribosome. Okay, then RER, then 2. So 231, what is it? 231, B option is right. So B option is right. Whoever has done the A wrong, that's very bad. I want to prepare for you. This is what you were before IMAT Accelerator, and now you have to improve it. Okay? I hope you guys are improving. So the following structures are found in plant cell. Cell wall is found in plant cell, cell membrane, nucleolus, mitochondria. Which cell structure might contain molecules containing carbohydrate monomers? Tell me, brothers, which one has got which one contains carbohydrate monomers? Hurry up. I hope they, I don't know. First of all, how many students like put it wrong? I don't know. Literally, I don't know. How many students have done it wrong? That's bad. Okay, that's bad. So I my fingers are like spinning. The following structures are found in plants. So they're going to be, this is like past paper question of I think 2018 or 2017. I think 2017 or 2018, whatever it is. But now cell wall contains carbohydrates, which is cellulose. Cell membrane also contains carbohydrates. Nucleolus, we are not sure about nucleolus because we haven't studied. But if we take nuclear membrane, it too contains. Okay, it also haven't written the nuclear membrane, but nuclear membrane does contains the carbohydrates. Then mitochondria has got cell membrane. It will be containing carbohydrate. So all options are supposed to be true. Okay, all options are supposed to be. Okay, let's move it. Okay, Maria, you haven't studied this. Let's go back. I wasn't seeing again. Okay, let's go back. Okay, so see here. Understand the question first of all. Which of the following transport mechanism require the use of proteins? Okay, use of proteins, molecules found in the membrane and ATP. I told you, I didn't told you about passive diffusion. This is supposed to be passive diffusion. Okay, we didn't. I told you passive diffusion is just diffusion, moving from higher potential to lower potential. It will not require protein. Okay, it doesn't require protein. But let me tell you, if you are saying protein is required, let's talk about active transport, diffusion, facilitated diffusion. Diffusion, if we say diffusion requires sort of protein, it does not require ATP. It is just simple passive diffusion. Passive diffusion is just movement from higher to lower potential without the use of ATP. But question is saying that is going to use ATP. Facilitated diffusion does not use ATP. So this is going to wrong. Diffusion is going to wrong because they not are using ATP. Active transport is going to use ATP. Active transport is active transport, lower potential to higher potential using of like ATP. So active transport is only the right option here. Are you understanding my with this question? Why it's wrong? Yeah, solution is same. The answer is marked wrong. Why, Loita? Why it's wrong? The D is true. Only Lohita, tell me if you're saying answer is marked wrong, why it also says A as wrong? Why it says A as true? I have taken it from past papers and I got the answer. I can like just go and chat GPT it. You will get the answer. Okay, let me scroll a bit. I am telling no, sorry, I have made it a wrong. Literally, I have put it wrong. Okay, sorry, I was like, I have made it a hurry. That's why I am telling you only option one is wrong, true. Okay, literally, it's my apologies. Okay, okay, really sorry. I told you answered the wrong. I didn't like look back at the sorry. Okay, sorry, everybody. Sorry for the. I already made it out. Like okay. I made it wrong. I didn't know. I haven't noticed it. Okay. I didn't like got to the correct right. I was like seeing D is the right and I was saying it right. I didn't know it. I have marked it A. Okay. Sorry. I'm really sorry. So I hope you guys got it because I explained 3 times. Diffusion is wrong. The diffusion octave transport is true. So option D is just true. So I didn't know A is wrong. Okay, sorry, sorry, correct. Sorry, I am really sorry. I hope you guys got this question. Now let's talk about this. Which organelle contains RNA but not DNA? It's very simple. It's very simple. Nucleolus contains DNA. Mitochondria contains DNA. Chloroplast contains DNA. Circular DNA. Circular DNA. It contains double-stranded DNA. Smooth endoplasmic reticulum does not contain DNA, but ribosome does not contain DNA. Okay, because smooth endoplasmic RNA also smooth endoplasmic also not includes RNA, but it's saying it also include RNA. But a ribosome contain RNA, but it does not contain DNA. Okay, smooth endoplasmic does not contain RNA as well as DNA. So ribosome is true. I hope you guys are getting it. And then you were saying no one go full. Sorry. Okay. I will update it. I will update the answer, and we will see how many students get the good option. Let me see. Let me update the answer. Let me update the answer, and then we can like see how many students got. Nobody got full marks either. Okay. Nobody got full marks either. Whoever what is saying, Dev, you were saying nobody. You were saying nobody got full. Okay. After updating the answer, nobody got full. Now I think you need to work on your like thing. Okay. Dev, nobody got full. Literally, those who were getting 2 marks, those got three marks. Okay, and rest all are like same. Like say more. Maybe somebody would have got full, and if I would have changed, then they would go. That didn't happen either. Yeah, you got 5. You didn't got 6. Okay, Rania, you are also 5. You are also 5. You have also got 5. Okay. Anyways, let's talk about it. That's for fun. You are also you have Rania. What you? Yeah. Now you have got 5. Okay. So I hope you guys got the answer. Now this was the end of the like session. So ribosome has got proteins and ribosome. I hope you got. Okay, that's good. That's good. Now this will appear. Now this was just past paper. Maybe somebody would have solved it earlier. Your real potential will come when you will solve mock exam. That would be 50 questions, 60 questions. Your real potential will come that time. And that student who didn't solve it, okay, he will not be allowed in classes. And second thing, he will be penalized. Okay, if he wanted to take class, he will have to pay some sort of certain. We will consecutively come up with a sort of a collective mm, a fine or maybe any penalty that we should do for that person, because now many students will flinch from mock exams. The mock exams are going to be like super helpful, and you will have to attempt it. I hope you guys got it. So this was today's session. So I want to hear you. Central question got me. Which central? Central question. Yeah, you have to be quick. Yo, in IMAT exam, you guys all have to open your ears, mind, eyes, bus, just that thing. Okay, not mouth. Okay, no mouth. Mouth should be quiet at your exam. Okay, otherwise examiner will kick you out from the exam hall. You just have to open up your ears. Also nose should be open. Okay, nose should be open. How you will be breathing. Okay, okay, Dev, that is specifically for you. Yeah, nose from where you will breathe. I hope you guys got it. How was today's session? Hopefully that was good. I don't know, but this is what we will be doing every day. So join every day on time, 10-15 minutes earlier, and we will have super super chill. sessions, and you will be doing lots of self-study. As I told you, self-study is very mandatory, so do pre-study earlier to IMAT exam. That will be super helpful. Okay,
so I hope you have enjoyed this video, and just let me know in the video description what are your thoughts, and the video and the notes, okay, from which I have explained, that will be in video description. You can download for free, and yeah, that's all. So just like it and subscribe this channel. See you in next video. Till bye bye.